Welcome to Audio Sleep Stories for Adults. Amidst the relentless rush of everyday life, there exists a tranquil space where profound ideas are gently woven into soothing narratives, inviting you to listen deeply while your mind drifts softly into peaceful slumber. Tonight, we invite you to journey with us into the mysterious world of immortal microorganisms, a silent realm thriving in invisibility, harboring secrets that challenge the very boundaries of life and time. Have you ever wondered what's truly happening all around you, unseen and unnoticed? Beneath the surface of the world you know, ancient life forms endure beyond the ordinary scales of existence. Among them, certain microorganisms possess a remarkable gift, an escape from death itself. Take, for instance, Turritopsis doanea asterisk, often called the immortal jellyfish. Unlike creatures bound by a single lifetime, this tiny being can revert its cells to an earlier stage, essentially pressing a biological reset button, allowing it to bypass aging and potentially restart its life cycle indefinitely. Imagine a creature capable of cycling through youth over and over, defying the natural order that claims all other life. Or consider Deinococcus radiodurans asterisk, a bacterium so resilient it laughs in the face of radiation levels thousands of times greater than humans can withstand. Its secret lies in a meticulous repair system, rapidly mending shattered DNA strands and restoring order where others succumb to chaos. As you settle deeper into this moment, breathe slowly and picture these microorganisms, invisible architects of survival, masters of regeneration, and ancient travelers through epochs of change. They exist in your body, your home, the soil beneath your feet, quietly rewriting the definitions of life, death, and time. Close your eyes and let their silent dance of immortality take you beyond the limits of ordinary perception, into a realm where life persists endlessly, and mysteries unfold within the tiniest of forms. Now softly, we step together into this extraordinary world. Imagine, for a moment, a creature so small that it slips through the mesh of the most delicate net, invisible to the naked eye, yet so resilient that it laughs in the face of time itself. Picture billions of these minuscule entities, nestled deep within the Earth's crust, thriving in scalding hydrothermal vents or the frozen abyss of Antarctic ice, defying the very limits we thought life must obey. What if I told you that these beings, these microorganisms, carry within them a secret whispered since the dawn of life, the secret of near immortality? This is not science fiction. This is the story of immortal microorganisms, the microscopic masters of survival whose essence challenges and reshapes our understanding of life, death, and the passing of time. Our journey begins not with textbooks or dry definitions, but with a glimpse into a world so alien, yet so intimately tied to our existence. Picture a brilliant microbiologist named Dr. Lynn Margulies, whose life was a symphony of curiosity and revelation. In the late 20th century, Margulies championed the theory of endosymbiosis, the idea that complex life itself arose from a partnership between distinct single-celled organisms. These tiny architects of life, the microorganisms, were not mere background players but pioneers wielding the power of resilience and transformation. In their story lies the heartbeat of immortality. To grasp this concept, one must first understand what immortal means in this microbial context. Unlike the human quest for eternal youth or physical immortality, immortal microorganisms do not evade death in a mystical sense. Rather, they possess mechanisms that allow their lineages to persist indefinitely, through cycles of dormancy, regeneration, and adaptation that blur the boundary between life and death. Consider Deinococcus radiodurans, famously dubbed Conan the bacterium for its near indestructibility. Discovered in the 1950s, this bacterium can endure doses of radiation that would obliterate most life forms, repairing its shattered DNA with a precision that defies biological logic. NASA's research in 2010 revealed that *D radiodurans could survive not just terrestrial hazards, but the vacuum and cosmic radiation of space itself, sparking speculation about life's potential voyages beyond Earth. The essence of microbial immortality is not magic. It is an evolutionary masterpiece encoded in DNA and expressed through biochemistry. These microorganisms employ multiple strategies: cryptobiosis, where metabolic processes grind to a halt as if time itself pauses; horizontal gene transfer, allowing them to swap survival tools like digital data packets; and biofilm formation, creating protective multicellular fortresses impervious to external assault. In the arid soils of the Atacama Desert or the depths of Siberian permafrost, dormant microbes have been revived after tens of thousands of years, essentially leaping across epochs. Their survival speaks to a form of immortality intertwined with time's elasticity, a biological principle that challenges our linear narrative of life and death. When we contrast immortal microorganisms with other forms of life, the distinction crystallizes. While trees may live for millennia and certain animals exhibit remarkable longevity, their mortality remains an unavoidable endpoint. Microbes, in contrast, exist as a vast network of genetic continuity. Their immortality is collective rather than individual. The entity that dies is the cell, but the lineage marches on, a perpetual relay race against entropy. Philosophically, this biological persistence invites reflection on the nature of self and identity. Are we to define an organism by the lifespan of its physical form, or by the continuity of its genetic heritage? What does it mean to be alive, when metabolic activity can cease for millennia, only to resume as if awakening from a cosmic slumber? The boundaries between life and death, individuality and community, persistence and extinction become porous in the microbial realm. This challenges human notions of uniqueness and permanence, nudging us towards a humbler, more interconnected vision of existence. Further enriching this narrative, consider the work published in Nature in 2023, where scientists uncovered microorganisms within rock formations deep beneath the ocean floor that appear to metabolize so slowly as to consume mere molecules of energy over centuries. These ultra-slow metabolisms suggest a form of life existing at the edge of what we define as life itself, bridging the gap between the animate and inanimate. They raise profound questions. Is life's defining characteristic the vigor of its metabolism, or the capacity to persist, adapt, and endure? In 2016, a remarkable study detailed bacteria revived from amber, frozen in time millions of years ago. These organisms did not merely survive, they thrived upon resurrection. Such findings emphasize the incredible plasticity of life, its ability to withstand cataclysmic shifts, and the relentless passage of time. It is as if these microorganisms hold in their molecular memory the chronicles of Earth's history, acting as sentinels that connect past, present, and future life. The interplay of such science with human imagination can be found in the hypotheses about panspermia, the theory that life on Earth might have extraterrestrial origins, hitchhiking on comets or meteorites. Immortal microorganisms, robust against space radiation and vacuum, become the cosmic travelers of life's saga, potentially seeding worlds billions of years apart. This idea stretches our contemplation beyond Earth, illuminating a universe where life is not a rare gem, but a tenacious principle woven into the fabric of the cosmos. Yet, amid this awe-inspiring resilience lies a paradox that stirs deep philosophical unease. If life can persist indefinitely in microscopic forms, what then of our human mortality? Does the continuity of microbial lineages diminish the significance of our individual fleeting existence, or does it enrich the tapestry into which each of us is woven? Here, the story of immortal microorganisms becomes a mirror reflecting our deepest existential questions, questions about legacy, meaning, and the nature of time itself. Modern science continues to unravel these mysteries. The advent of CRISPR gene editing and synthetic biology now allows us to glimpse how we might harness or mimic these natural strategies for longevity and repair. The 2019 research into extremophiles, organisms thriving in conditions once thought uninhabitable, pushes us to reconsider the limits of life and the possible futures of biotechnology. Could immortal microorganisms inspire breakthroughs not only in medicine, but in redefining what it means to be alive? As we stand on the threshold of these revelations, the story of immortal microorganisms unfolds as both a scientific odyssey and a philosophical inquiry. It is a story of resilience, continuity, and the tenacity of life in the face of oblivion. It reminds us that immortality is not merely about eternal life, but about the persistence of identity, memory, and existence across the vast canvas of time. So, as we peer deeper into the microcosm, witnessing these tiny immortals thriving against cosmic odds, we are invited to ponder. In a universe where life can endure in whispers and shadows, where does that leave our human story? So brief, so fragile, yet so intensely alive. What does immortality mean to us when the smallest creatures already hold it in their microscopic hands? Delving deeper into the essence of immortal microorganisms, it becomes imperative to explore the molecular and cellular mechanisms that confer their extraordinary resilience. One of the most remarkable features enabling microbial immortality is the ability to maintain and repair genomic integrity under conditions that cause severe DNA damage. For instance, Deinococcus radiodurans can repair up to 200 double-strand breaks per genome, a degree of damage lethal to most cells, within hours after radiation exposure. This process is orchestrated by a complex network of repair enzymes, including highly efficient homologous recombination pathways and unique proteins such as PPRA, which protects and stabilizes broken DNA ends. The bacterium's genome architecture also contributes, with multiple copies of its genome polyploidy, providing templates for accurate repair, ensuring the survival of its lineage. These findings are supported by studies from scientists like Dali et al. 2007, who elucidated the robust mechanisms underlying *D. Radiodurin's resistance to oxidative stress and ionizing radiation. Moreover, the phenomenon of cryptobiosis, derived from Greek words meaning hidden life, is another key survival strategy that extends microbial lifespans beyond conventional boundaries. This state, characterized by an almost total suspension of metabolic activity, allows microorganisms to withstand extreme desiccation, freezing, and oxygen deprivation. Tardigrades, although multicellular and not microorganisms per se, famously exemplify cryptobiosis, but many bacteria and archaea utilize variants of this strategy. For example, bacterial endospores formed by species like Bacillus subtilis can survive in a dormant state for millions of years, resisting heat, radiation, and chemical insults due to an extraordinarily resilient spore coat and DNA repair machinery activated upon germination. Research published in Geobiology, Nicholson et al. 2000, highlights the potential longevity of spores and their implications for understanding life's persistence across geological timescales. Horizontal gene transfer, HGT, introduces an additional dimension to microbial immortality by enabling the rapid acquisition of novel genetic traits that enhance survival. Unlike vertical gene transfer, which relies on reproduction, HGT allows organisms to exchange genetic material across species boundaries, effectively sharing evolutionary innovations in real time. Plasmids, transposons, and bacteriophages serve as vehicles for this genetic exchange, facilitating adaptation to new environmental stresses. This dynamic genetic fluidity ensures that microbial populations remain not only resilient but highly adaptable, counteracting the deleterious effects of mutations and environmental upheavals. Seminal work by Frost et al. 2005, in Nature Reviews Microbiology, extensively catalogs these processes and their role in microbial evolution. Biofilms represent a fascinating multicellular-like behavior in microorganisms, where cells aggregate within a protective extracellular matrix composed of polysaccharides, proteins, and nucleic acids. This communal living arrangement affords microbes significant protection from antibiotics, desiccation, and immune responses, thereby enhancing survival in hostile environments. Within biofilms, horizontal gene transfer events are especially prevalent, further promoting adaptability and persistence. A review by Hall-Studley et al. 2004, in Nature Reviews Microbiology, underscores the importance of biofilms in chronic infections and environmental resilience, illustrating how this communal lifestyle contributes to the concept of microbial immortality. Another facet of microbial immortality lies in ultra-slow metabolism and energy conservation strategies. Studies probing microbes inhabiting subseafloor sediments, such as the work published in Proceedings of the National Academy of Sciences, DeHaan et al., 2019, reveal organisms metabolizing at rates so low that they consume energy equivalent to a few molecules annually. These cells maintain vital functions like DNA repair and membrane integrity at near-threshold levels of energy throughput, effectively existing on the edge of life. Such minimal metabolism blurs the line between dormancy and activity, expanding our understanding of what constitutes a living organism. This life at the edge exemplifies an alternative model of immortality, sustaining viability across immense timescales through biochemical minimalism. From an evolutionary standpoint, immortal microorganisms provide a living record of Earth's biological history. Genomic analyses uncover conserved sequences and ancient gene families that trace back billions of years, suggesting that these resilient forms may closely resemble some of the earliest life on Earth. The discovery of microbial dark matter, lineages identified only through environmental DNA sequencing, has expanded the tree of life and revealed previously unknown taxa thriving in extreme habitats. Studies such as those by Hug et al. 2016 in Nature Microbiology highlight the vast diversity and evolutionary significance of these uncultured microorganisms. These findings not only emphasize their role as preservers of genetic heritage, but also as active participants in biogeochemical cycles essential to planetary homeostasis. In the realm of astrobiology, the resilience of immortal microorganisms has profound implications. Their capacity to survive vacuum, intense radiation, and temperature extremes makes them primary candidates for interplanetary transfer of life via panspermia. Experiments conducted aboard the International Space Station, such as those reported in Astrobiology 2019, have demonstrated that spores of Bacillus species and other extremophiles can endure prolonged exposure to space conditions. These results strengthen hypotheses that life could theoretically traverse cosmic distances, potentially seeding habitable niches on distant worlds. The forthcoming ESA mission, ExoMars, aims to detect such potential microbial life or biosignatures on Mars, informed by insights from Earth's immortal microorganisms. Biotechnological applications emerging from our understanding of microbial immortality are equally promising. DNA repair enzymes from D-radiodurans are under investigation for enhancing gene therapy and cancer treatments by improving cellular resistance to radiation. Similarly, biomolecules involved in cryptobiosis inspire the development of novel preservation techniques for pharmaceuticals and biological samples. The field of synthetic biology seeks to engineer microorganisms with tailored longevity traits, potentially revolutionizing bioremediation and industrial processes. For example, the CRISPR-Cas systems, originally discovered as bacterial immune mechanisms, now serve as transformative gene editing tools, illustrating how knowledge of microbial survival strategies can translate into groundbreaking technologies. Recent reviews in Trends in Biotechnology, 2022, discuss these advances and their future trajectories. Philosophically, immortal microorganisms compel us to reconsider foundational concepts in biology and metaphysics. The classical notion of individuality becomes fluid when confronted with microbial communities, exchanging genes and sharing protective matrices. The persistence of genetic material beyond cellular death challenges the finality of mortality, suggesting a continuum rather than a dichotomy of life and death. This resonates with indigenous and Eastern philosophies that emphasize interconnectedness and cycles rather than linear progression. The biologist and philosopher Francisco Varela proposed the concept of autopoiesis, self-producing systems, to describe living organisms, a framework that accommodates the dynamic, networked nature of microbial life. Such perspectives encourage a more holistic view of life's persistence, one that transcends the human-centric narrative. In conclusion, the nature and definition of immortal microorganisms transcend conventional biological categories. Their resilience arises from a confluence of molecular mastery, metabolic strategy, communal living, and evolutionary legacy. These organisms epitomize life's tenacity, illustrating that immortality in the biological realm is less about eternal individual existence and more about the perpetual continuity of genetic and metabolic processes. They invite us to reflect on the profound interconnectedness of all life forms and the subtle, enduring dance between survival and transformation. As we continue to explore their mysteries, immortal microorganisms stand as both scientific marvels and profound symbols of life's boundless capacity to endure. The flickering flame of a candle cast long shadows across the cluttered desk where Alexander Fleming, the Scottish bacteriologist, sat hunched in the solitude of his laboratory. It was the dawn of the 20th century, an era brimming with both scientific triumphs and the lingering shadows of ancient mysteries. Yet, somewhere beneath the swirl of Petri dishes and microscopes, a profound question had begun to take root in the minds of a few daring souls. Could some forms of life, these tiny architects of biology, evade the finality of death? Could microorganisms, creatures invisible to the naked eye, possess a secret to immortality, a defiance of decay that challenges the very definition of life and death itself? This question, tenuous and almost heretical at the time, whispered through the annals of science, philosophy, and mysticism, evolving from obscure notes in dusty manuscripts to heated debates in the hallowed halls of academia. The history of immortal microorganisms is not merely a chronicle of discovery. It is a saga of human curiosity grappling with the boundary between the ephemeral and the eternal, the known and the unknowable. Our story begins long before Fleming's serendipitous discovery of penicillin, reaching back to antiquity, when civilizations first stared into the abyss of decay and disease, and wondered if life could, in some way, overcome its own end. Ancient Indian texts, such as the Atharva Veda, reference Amrita, the nectar of immortality, believed to grant eternal life. While not explicitly about microorganisms, these mythic aspirations reflected a profound, universal yearning to transcend mortality. It would take millennia before this longing would converge with the tangible, microscopic world. Fast forward to the 17th century. A dimly lit chamber illuminated by candlelight reveals Antony van Leeuwenhoek peering through his handcrafted lens. In 1676, the Dutch tradesman turned scientist spotted, for the first time, the animalcules, bacteria and protozoa swimming in a drop of pond water. Here, humanity glimpsed the hidden universe within, a realm where life thrived invisible to the naked eye. This revelation unleashed a cascade of questions. How do these microorganisms live and die? Could any possess a resistance to time's ravages? Yet, the idea of immortal microbes seemed absurd in the era dominated by the miasma theory of disease and the religious belief that death was the ordained end of all living beings. The concept quietly simmered in the background, gathering steam only centuries later as microbiology matured into a rigorous science. The 19th century brought the germ theory and groundbreaking minds like Louis Pasteur and Robert Koch, whose meticulous experiments unveiled the roles microbes played in fermentation and disease. However, amidst their successes lurked unexplained phenomena. Certain bacterial spores seemed indomitable. They resisted boiling, survived harsh chemicals, and remained dormant for decades, even centuries. Were these mere survival mechanisms or glimpses of a deeper biological enigma, quasi-immortality? One of the earliest and most famous examples documenting this resilience was the case of bacillus spores found in ancient tombs and salt crystals. In the 19th century, reports emerged of viable bacteria isolated from Egyptian mummies and 2-million-year-old salt deposits. These claims, often met with skepticism, spurred spirited debate. Could life truly persist unchanged over geological epochs? Enter John Tyndall, the Victorian physicist obsessed with sterilization, whose experiments in the 1870s challenged and refined the understanding of microbial endurance. Tyndall observed that some spores resisted heat unless exposed for extended periods, coining the term heat-resistant spores. Though he did not claim immortality, his work laid a foundation for questioning microbial mortality's absoluteness. Meanwhile, in the spiritualist and pseudoscientific circles of late 19th century Europe, a different narrative unfolded. Ideas about etheric life forces and spiritual microorganisms animated by some life energy captivated public imagination. This cultural crosscurrent underscored the fraught interplay between science and metaphysics that would shadow the discourse on immortal microbes for decades. The dawn of the 20th century offered a new protagonist in this narrative, Vladimir Vernadsky, the Russian mineralogist and biogeochemist. Vernadsky proposed the concept of the biosphere, emphasizing life's profound influence on the planet's geology and chemistry. In his vision, microorganisms were not transient flickers, but foundational and enduring agents in Earth systems. Could this scientific view accommodate the possibility that certain microorganisms might, in essence, live forever by virtue of their capacity to persist and evolve through extreme conditions? This question gained urgency during the Cold War, when the scientific community's gaze turned toward extremophiles, organisms thriving in boiling acid pools, frozen permafrost, and radioactive waste. Discoveries by Thomas Brock in the 1960s, isolating Thermus aquaticus in Yellowstone's hot springs, revolutionized biology and hinted at life's extraordinary adaptability. These extremophiles raise the tantalizing possibility that some microorganisms embody a form of biological immortality, not by avoiding death, but by entering suspended animation impervious to time's decay. Parallel to this Western scientific journey, the East nurtured its own traditions of microbial immortality, albeit framed within holistic philosophies rather than empirical science. Traditional Chinese medicine, for instance, embraced the concept of qi, an energy flow sustaining life, and certain herbal concoctions were believed to nurture longevity at the cellular and spiritual levels. While not directly addressing microorganisms, this worldview embedded a cultural acceptance of life's cyclical renewal and persistence, a subtle nod to microbial resilience as a metaphor for immortality. The latter half of the 20th century was marked by intense academic controversy. The discovery of bacterial DNA repair mechanisms, like those in Deinococcus radiodurans, famously dubbed Conan the bacterium for its near indestructibility, challenged the finality of microbial death. Scientists such as Michael Daly uncovered how these microbes could reconstruct shattered genomes, surviving radiation doses lethal to other life forms. These findings rocked the foundations of biology, forcing a reevaluation of death's universal dominion. Yet, the idea of immortality remained contentious. Critics argued that such microbes merely postpone death, entering dormancy or repair modes rather than achieving literal immortality. The line between survival and immortality blurred, sparking fierce debates, some echoing long-standing philosophical questions about what it means to be alive or dead. In academic papers like the 1998 publication in Science by Battista et al., the nuanced mechanisms of microbial resilience were meticulously documented, fueling both scientific veneration and skepticism. On the cultural front, the theme of immortal microorganisms fed into speculative fiction and popular culture. Writers like H.G. Wells and later Michael Crichton imagined scenarios where microbes defied death or even conferred immortality, weaving science with myth and exploring humanity's anxieties about aging, death, and the limits of life. These narratives, while fictional, reflected and amplified public fascination with the concept, often prompting real scientists to clarify and investigate the boundaries of microbial longevity. Today, the story continues in the form of deep subsurface biosphere studies and astrobiology missions seeking life beyond Earth. Microorganisms discovered thousands of meters beneath the planet's crust, surviving on mineral energy alone, suggest that microbial life in some form has persisted for billions of years, arguably the closest embodiment of biological immortality known. NASA's Perseverance rover, scouring Mars's ancient lake beds for biosignatures, extends this quest beyond our planet, challenging us to redefine life's tenacity and perhaps its immortality. Through this winding, century-spanning journey, one tangible relic remains, a spore sample preserved at the American Type Culture Collection, originally isolated in 1965 from a desert soil crust. It is a silent witness to debates, experiments, and aspirations. Stored in liquid nitrogen, it awaits the curious eyes of future scientists who may yet unravel the final secrets of microbial survival. And so, as we stand at the crossroads of knowledge and mystery, we must ask, if some microorganisms can evade death for millennia, repairing and renewing themselves, what does that mean for the nature of life itself? Is biological immortality a threshold we can cross, or an eternal horizon forever just beyond our reach? This question beckons us onward, into the depths of today's hypotheses and controversies, where science and philosophy entwine once again. As the 20th century progressed, the increasing sophistication of molecular biology and genetics provided a powerful lens through which to view microorganisms' survival capabilities. The intricate processes governing DNA replication, repair, and mutation became clearer, revealing that certain microbes had evolved incredibly efficient mechanisms to counteract the damage brought on by environmental stressors. The seminal work of scientists like Joshua Lederberg and Matu Kimura in the mid-1900s helped to establish the molecular underpinnings of microbial robustness, showing that evolutionary pressures had shaped microbes not only to survive, but to thrive under conditions that would obliterate most multicellular life. One notable breakthrough emerged from the study of bacterial endospores. These unique, dormant structures produced by genera such as Bacillus and Clostridium can withstand extreme heat, desiccation, radiation, and chemical assault. Investigations by Howard Stroll and colleagues in the 1970s dissected the biochemical composition of spores, revealing protective layers of dipicolinic acid and small acid-soluble spore proteins, SASPs, that shield DNA from damage. These findings reinforce the idea that spores serve as biological time capsules, allowing microbial life to pause its metabolic processes and reawaken when conditions improve, a form of suspended animation rather than true immortality, but nonetheless a remarkable survival strategy. The concept of cryptobiosis arose to describe such reversible states of extreme metabolic inactivity. Microorganisms entering cryptobiosis can halt life processes to near undetectable levels, then resume normal function, potentially after centuries or millennia. In 1995, biologist William B. Miller, Jr., extensively cataloged cryptobiotic organisms across various taxa, noting the astounding parallels between microbial spores and other life forms such as tardigrades and nematodes. While cryptobiosis is not exclusive to microbes, it underscores the broader biological principle that life can adopt states indistinguishable from death, challenging traditional definitions of mortality. Beyond Earth's confines, the burgeoning field of astrobiology has fueled interest in microbial longevity with the hypothesis that life forms, if they exist elsewhere in the cosmos, must possess extraordinary resilience. Experiments exposing bacteria to the vacuum and radiation of space, such as those conducted aboard the European Space Agency's BIOPAN and EXPOSE missions, demonstrated that certain spores could survive for years under extraterrestrial conditions. In 2007, the Tanpopo mission aboard the International Space Station successfully detected viable microbes after prolonged exposure to space, lending credence to models like panspermia, which propose that life can travel between planets via meteorites. While not proof of immortality, these findings suggest microorganisms can endure the interplanetary journey of millions of years, effectively sidestepping death across cosmic timescales. The late 20th and early 21st centuries also saw the emergence of epigenetics and systems biology, further complicating the picture of microbial death and survival. Epigenetic modifications, heritable changes in gene expression without changes to the DNA sequence, play crucial roles in microbial adaptation to environmental stress. Studies led by researchers such as Susan Lindquist at MIT uncovered how microbial populations can remember stressful encounters and adjust accordingly, enhancing survival rates over generations. This molecular plasticity blurs the line between death and survival, implying that microbial life is not a fixed state, but a dynamic process capable of near-continuous renewal. Importantly, the debate over microbial immortality has also been shaped by technological advances in metagenomics and single-cell sequencing. These methods have unveiled the vast diversity of unculturable microbes residing in extreme environments like deep oceans, subterranean rocks, and polar ice. For instance, the identification of ultramicrobacteria in oligotrophic nutrient-poor environments highlights microbial minimalism as a strategy for longevity, with some cells maintaining ultra-low metabolic rates for extended periods. A 2016 study published in Nature Communications by Lever et al. Revealed bacterial communities persisting in million-year-old subseafloor sediments, existing at metabolic rates millions of times slower than surface microbes. Though these cells are metabolically active, their energy demands are so minimal that questions arise. Is their lifespan effectively extended? And if so, does this approach biological immortality? Parallel to empirical discoveries, theoretical biology grappled with the philosophical implications. The concept of the immortal germ line, first proposed by August Weismann in the late 19th century, posited that while individual organisms age and die, the genetic material passed through successive generations is effectively immortal. Modern interpretations of this theory consider microbial populations as perpetually renewing lineages, each cell a potential progenitor, thus embodying a form of biological continuity that transcends individual mortality. This framework has found support in the work of biogerontologists studying senescence and cellular aging, suggesting that microbial immortality is less about indefinite survival of one cell and more about the unbroken transmission of genetic information. Critically, recent research into microbial community resilience and biofilms adds another dimension. Biofilms, complex assemblies of microbes embedded in self-produced extracellular polymeric substances, offer collective protection from antibiotics, desiccation, and immune system attacks. Studies led by J. William Costerton in the 1990s and onwards have shown that biofilms behave as superorganisms, displaying emergent properties that exceed the sum of their parts. Within biofilms, dormant persister cells can withstand hostile environments for prolonged periods, reactivating later to repopulate. This collective survival strategy challenges traditional microbiological paradigms and suggests that microbial immortality might be viewed not solely at the cellular level, but as a property of communities. In the contemporary era, synthetic biology and genetic engineering have begun to explore the possibility of designing microorganisms with enhanced survival traits or controlled lifespans. Efforts to engineer bacterial strains capable of programmed cell death or self-repair could redefine notions of microbial life cycles and potentially longevity. A seminal 2013 paper in Nature by Liu et al. Demonstrated synthetic gene circuits that precisely regulate bacterial population dynamics. While these interventions do not create immortal microbes, they represent a profound step toward understanding and manipulating microbial persistence. Moreover, the environmental and medical significance of microbial longevity cannot be overstated. Persistent pathogens capable of evading host defenses and antibiotics, such as Mycobacterium tuberculosis or Staphylococcus aureus persister cells, pose major public health challenges. Understanding the molecular basis of their prolonged survival informs treatment strategies and underscores the practical importance of microbial resilience research. In conclusion, the history of microbial immortality is one of evolving understanding, from mystical speculations to rigorous scientific inquiry. The concept remains nuanced, resisting simplistic definitions. Microorganisms have developed astounding mechanisms to survive extreme conditions and extended time periods, but whether this constitutes true immortality depends on how one frames life, death, and continuity. As new tools and theories emerge, the boundary between ephemeral life and enduring existence continues to be redrawn, inviting us to reconsider the essence of life itself in the microscopic world. In a dimly lit lecture hall at a prestigious international microbiology conference, the air buzzes with anticipation. Scientists from across the globe gather, their faces illuminated by the glow of a massive blackboard covered in intricate diagrams of cellular structures and genetic sequences. At the center, Dr. Evelyn Hartman, a virologist with a reputation for challenging orthodoxies, adjusts her glasses and begins her presentation. She poses a question that has stirred debate for decades. Could microorganisms possess a form of biological immortality? This question, seemingly plucked from the realm of science fiction, serves as a portal into a labyrinth of hypotheses, rivalries, and intellectual duels that have shaped our understanding of life's resilience. The story of immortal microorganisms begins in the mid-20th century, where the first hypothesis finds its roots in the observations of extremophiles, microbes thriving in boiling hydrothermal vents miles beneath the ocean's surface. Dr. Thomas W. Calder, a marine biologist fascinated by life in Earth's harshest environments, popularized the concept of biological stasis in the 1960s. Calder proposed that certain microorganisms, like the archaea from deep-sea vents, achieve a near-immutable state, not by evading death through rapid replication, but by slowing their metabolism to an almost imperceptible crawl. This metabolic suspension theory was grounded in studies published in Nature, Calder, 1968, where he demonstrated that some thermophiles survived prolonged exposure to sterilizing conditions that annihilated most life forms. Calder's hypothesis brought the marine world to the forefront of immortality debates, suggesting that life's persistence isn't necessarily a race against time but a strategic retreat into dormancy. However, this notion sparked fierce opposition. Critics, led by Dr. Sandra Millington of MIT, argued that metabolic slowdown was a form of suspended animation rather than true immortality. In her 1973 science paper, Millington posited that these microbes do not escape death, they merely delay it. To her, immortality requires continuous self-repair and replication without senescence, a process she felt archaea failed to demonstrate unequivocally. The debate escalated beyond biology into philosophical terrain. If a microorganism simply pauses life, is it truly immortal or just in a state of limbo? This question echoed the famous paradox in astrophysics of a black hole's event horizon, where time seems to stop for an outside observer. Popular culture mirrored this quandary. The cybernetic organisms of Blade Runner and the time-warped consciousness in Black Mirror episodes evoked similar themes of suspended existence versus authentic immortality. Calder's camp found supporters in evolutionary biologists fascinated by life's ability to endure in stasis, while Millington's followers favored a more dynamic vision of immortality. Parallel to this debate emerged the second prominent hypothesis in the 1980s, inspired by discoveries of bacterial spores exhibiting extraordinary longevity. Dr. Harold Finch, a microbiologist at the University of Arizona, championed what became known as the genetic repair immortality hypothesis. Finch's team studied Bacillus subtilis spores extracted from salt crystals dated at over 250 million years old, findings partially endorsed by NASA's Astrobiology Division in a 1994 *PNAS* article. According to Finch, these spores were not merely dormant but possessed molecular mechanisms enabling continuous DNA repair, even in an inert state. This repair machinery, theoretically, could counteract the cumulative mutations that inevitably lead to cellular death. Finch's hypothesis was revolutionary. It proposed that immortality might lie not in avoiding damage but in perpetual molecular repair, a self-maintaining system akin to a cosmic phoenix rising eternally. His laboratory experiments demonstrated that under controlled hydration, these spores could repair decades-old DNA lesions and resume replication, suggesting a form of biological reboot. The implications caught the imagination of scientists and science fiction writers alike, inspiring narratives of microbial sentinels guarding secrets of eternal life in the far reaches of space. Yet Finch's vision faced its own skeptics. Detractors highlighted the improbability of perfect molecular repair over geological timescales, citing stochastic damage and entropic decay as insurmountable barriers. Dr. Alyssa Romano, a geneticist from the European Molecular Biology Laboratory, published a scathing critique in Cellular and Molecular Life Sciences, 1999, questioning the veracity of such long-term DNA integrity claims. Romano argued that even the most robust repair enzymes have finite fidelity and energy requirements, making immortality impossible without infinite resources. At this crossroads of biology and thermodynamics, the intellectual battlefield shifted toward energy economics, a third hypothesis that tied immortality to energy sustainability within microorganisms. Dr. Naoki Yamashita, a bioenergeticist from Tokyo University, advanced what came to be known as the minimal energy immortality model. Yamashita's insight was brilliantly simple. Immortality in microorganisms depends on an ultra-efficient energy economy that minimizes damage from metabolic processes themselves. His 2005 study published in PNAS showed that certain extremophilic microbes exhibit metabolic pathways producing near-zero reactive oxygen species, ROS, notorious culprits in aging and cellular damage. Yamashita proposed that these microorganisms achieve immortality not by halting metabolism or repairing damage endlessly, but by producing negligible metabolic byproducts that cause harm. This model shifted the conversation from DNA repair systems, or dormancy, to the quality and efficiency of bioenergetics at the cellular level. His work was bolstered by quantum biology research from MIT, exploring how electron transport chains in some microbes utilize quantum coherence to reduce energy wastage, a notion that sounded like science fiction, but was increasingly grounded in experimental evidence. The minimal energy immortality hypothesis garnered broad interest but also skepticism, particularly from ecologists who emphasized environmental unpredictability. They argued that even the most efficient energy usage could not guarantee immortality if external stressors, radiation, predation, chemical insults, intervene. This led to lively debates at conferences, often culminating in heated exchanges reminiscent of courtroom dramas, where each side presented meticulous evidence and counterarguments. A fascinating twist in the narrative arrived in the form of a heterodox hypothesis, emerging from a small, independent research collective known as the Ian Project, based in a converted warehouse in Berlin. The team, blending microbiology, art, and speculative fiction, proposed the symbiotic immortality model. Led by Dr. Leela Kaminsky, a microbiologist and conceptual artist, they suggested that immortality might not reside in individual microorganisms, but in their networked symbioses, a collective consciousness preserved through horizontal gene transfer and communal repair mechanisms. Kaminsky's provocative project found its initial inspiration in the microbial mats of microbialites and their communal resilience, documented in a 2012 Science Advances paper showing extensive horizontal gene transfer, enabling rapid adaptation. The Ian Project's experiments involved creating synthetic microbial communities that shared genetic material freely, hypothesizing that such interconnectedness might amount to a form of emergent immortality. No single cell lives forever, but the system as a whole persists indefinitely. This hypothesis, straddling the boundary between biology and metaphysics, attracted both fascination and derision. Traditional microbiologists criticized it as metaphorical rather than empirical, while philosophers praised its challenge to the individualistic paradigm of life and death. The symbiotic immortality model found a cultural echo in science fiction series like Star Trek Asterisk, where collective consciousnesses such as the Borg raised questions about identity and mortality beyond the individual. As this intellectual drama unfolds, it becomes clear that the quest to understand immortal microorganisms does more than stretch our scientific paradigms. It forces us to reconsider what it means to be alive, to endure, and to transcend. From Calder's thermophilic metabolic suspension to Finch's indefatigable DNA repair, from Yamashita's quantum bioenergetics to Kaminsky's communal symbiosis, each hypothesis embodies a different facet of nature's enigmatic relationship with time and death. One might ask, does the truth lie in a synthesis of these views? Could microorganisms leverage metabolic suspension, continuous repair, energy efficiency, and symbiotic networks in concert to achieve a form of immortality far more complex than any single model suggests? This possibility is not mere speculation. Recent advances in metagenomics, quantum biology, and synthetic biology hint at an integrated system where immortality emerges not from isolation but from dynamic interplay. As Dr. Hartman closed her presentation, leaving the audience suspended between awe and skepticism, she posed a final, tantalizing question. If microorganisms can unlock the secrets to biological immortality, what might this mean for humanity? Can our understanding of these microscopic immortals guide us toward extending life, curing disease, or even transcending our biological limits? The question lingers like a cosmic riddle, inviting the next generation of thinkers to dive deeper into the microbial abyss where immortality both hides and beckons. The story of immortal microorganisms is not merely the story of tiny life forms defying death. It is the grand narrative of survival, identity, and the infinite dance between life and time. Building upon the rich tapestry of hypotheses concerning immortal microorganisms, recent empirical studies have begun to blur the boundaries among these models, suggesting that the phenomenon of microbial immortality may indeed be multifactorial and context-dependent. A pivotal contribution came from a 2018 study published in Nature Communications by a collaborative team from the Max Planck Institute for Marine Microbiology and the University of Tokyo. This research delved into the survival strategies of Thermococcus gamma-tolerans asterisk, an archaeon notable for its extraordinary resistance to ionizing radiation. The team uncovered a hybrid mechanism integrating metabolic suspension with enhanced DNA repair capabilities. Specifically, T. Gamma-tolerance reduces its metabolic rate drastically under radiation stress, a nod to Calder's metabolic suspension theory, but simultaneously upregulates a suite of robust DNA repair enzymes, including homologs of RECA and photolyase, thereby embracing Finch's genetic repair immortality model. This synergy allows the organism not only to pause damage accumulation, but actively reverse it upon returning to more favorable conditions. This finding is significant because it empirically demonstrates that metabolic stasis and molecular repair are not mutually exclusive, but can operate synergistically. Moreover, the study employed transcriptomic and proteomic analyses, revealing that the energy demands of repair processes are met through ultra-efficient electron transport chains optimized to minimize reactive oxygen species (ROS) production, aligning closely with Yamashita's minimal energy immortality concept. Such integration implies that an organism's bioenergetics are dynamically tuned to balance energy allocation between maintenance, repair, and survival, rather than prioritizing one mechanism exclusively. In parallel, advances in single-cell genomics and high-throughput metagenomics have provided new insights into microbial communities, particularly those inhabiting extreme environments like permafrost sediments and deep subsurface ecosystems. A groundbreaking paper in ISME Journal, 2020, led by Dr. Mariana Silva, characterized microbial consortia in 1.5 million-year-old Siberian permafrost, revealing extensive horizontal gene transfer (HGT) events that maintain genetic diversity and repair capabilities across species boundaries. This supports Kaminsky's symbiotic immortality hypothesis by demonstrating that gene flow in microbial communities can act as a communal genetic cache, buffering against deleterious mutations and environmental stresses. Further experimental evidence from synthetic biology corroborates this communal aspect. A 2021 study in Cell Asterisk engineered synthetic microbial consortia capable of exchanging plasmids encoding DNA repair genes, conferring increased lifespan and stress resistance compared to monocultures. These engineered communities exhibited emergent properties akin to a distributed immortality system, where the network as a whole maintains functionality despite individual cell turnover. Such results underscore the ecological validity of symbiotic immortality and hint at evolutionary pressures favoring cooperative strategies as a means of persistent survival. However, these hypotheses also grapple with fundamental thermodynamic constraints. The second law of thermodynamics stipulates that biological systems must expend energy to maintain order and counteract entropy. While the minimal energy immortality model offers a mechanism to reduce internal damage, it cannot create energy ex nihilo. Thus, environmental energy flux remains a critical factor. Extremophiles inhabiting energy-poor niches, such as deep subsurface lithoeutrophs, survive for millennia with extraordinarily low metabolic rates, but this implies that their form of immortality is conditional and probabilistic rather than absolute. Intriguingly, recent work in quantum biology has provided tantalizing clues about how microorganisms might surmount some energetic limitations. A 2022 *PNAS* article detailed how photosynthetic bacteria utilize quantum coherence to optimize energy transfer within their reaction centers, reducing energy loss and enhancing repair efficacy. though this phenomenon has not yet been linked directly to longevity or immortality. It suggests a possible evolutionary pathway for microorganisms to refine their bioenergetic efficiency to unprecedented levels. If such quantum effects extend to DNA repair or metabolic regulation, as hypothesized by Yamashita and colleagues, they could represent a crucial component in the puzzle of microbial immortality. Philosophically, the intertwining of these models challenges classical distinctions between life and death. Instead of a binary state, microbial immortality may represent a spectrum of survival strategies negotiated between metabolism, repair, and ecological interactions. This continuum echoes the concept of biological persistence, rather than absolute immortality, reframing our understanding of life's temporal boundaries. From a biotechnological perspective, unraveling these mechanisms holds immense promise. Insights into microbial DNA repair pathways have already informed cancer research, with enzymes like photolysis being explored for therapeutic applications in human skin repair. The potential to harness microbial metabolic efficiency and communal genetic exchange opens avenues for engineering robust synthetic cells and novel bioproducts capable of withstanding extreme conditions, valuable for space exploration and industrial biotechnology. In conclusion, the mosaic of hypotheses on immortal microorganisms, metabolic suspension, genetic repair, minimal energy metabolism, and symbiotic networks are increasingly seen not as competing doctrines, but as interconnected facets of a complex biological reality. The frontier now lies in dissecting how these strategies coalesce dynamically across diverse taxa and environments, enabling life forms to skirt the edges of mortality. The quest continues, a profound inquiry into nature's capacity not merely to endure, but to transcend the inexorable flow of time. Imagine a small laboratory tucked away in the bustling biotech hub of Boston, where Dr. Emily Chen, a passionate microbiologist with a relentless curiosity about life's resilience, first held a vial containing a strain of bacteria unlike any she had seen before. These microbes were not just surviving, they seemed impervious to the usual cellular decay and environmental stresses that doomed their cousins. This was the beginning of a journey that would change the way humanity perceives life, longevity, and the microscopic world around us. Emily's team had stumbled upon a strain of Danacoccus radiodurans, often dubbed the immortal microorganism, due to its extraordinary resistance to radiation, desiccation, and oxidative stress. Initially found in the radioactive waste sites, this microbe's survival capabilities felt almost otherworldly. When Emily introduced it into a series of experiments aimed at bioremediation, the process of using living organisms to clean polluted environments, she witnessed something remarkable. The bacteria thrived where others perished, breaking down toxic compounds efficiently without succumbing to their harmful surroundings. The first users who applied these bacteria to contaminated sites, including communities near nuclear disaster zones, expressed awe and cautious optimism. Local environmental engineers reported a 70% reduction in soil toxins over 6 months, a turnaround they hadn't thought possible without hazardous chemical interventions. But this was only the beginning. The implications of immortal microorganisms leaped far beyond Emily's initial vision. In medicine, these microbes became living blueprints for next-generation therapies. For patients with chronic wounds, particularly those suffering from diabetes, the application of specially engineered immortal bacteria accelerated healing by producing protective biofilms that resisted infection and promoted tissue regeneration. Clinical trials in Europe showed that such treatments reduced healing times by nearly 40%, saving countless limbs from amputation. Stories surfaced from patients like Marta, a 58-year-old diabetic woman from Barcelona, who described the treatment as nothing short of miraculous. For years, I battled wounds that refused to close. After the treatment, it felt like my body remembered how to heal. On the frontier of space exploration, immortal microorganisms redefined humanity's reach. NASA, recognizing the resilience of these tiny warriors, funded projects to incorporate them into life support systems aboard the International Space Station and planned missions to Mars. These bacteria were not just surviving radiation. They were cleaning air and water, recycling waste, and even synthesizing nutrients in microgravity. The Microbial Ecology in Space mission recorded a 25% increase in bioreactor efficiency when using asterisk Danacoccus species compared to traditional strains. This resilience offered a lifeline for long-term space travelers, transforming the vision of a self-sustaining extraterrestrial colony from science fiction into a plausible reality. In the realm of materials science, the unique biochemical pathways of immortal microorganisms inspired revolutionary advances. By unraveling how these bacteria repaired their DNA countless times without error, scientists developed novel polymers and coatings that mimic their protective mechanisms. The result: paint and materials able to self-heal cracks and resist environmental damage over decades. Japan led the charge with companies like Nippon Advanced Materials, releasing self-repairing infrastructure textures that reduced maintenance costs by 35% over 5 years. Cities employing these innovations reported fewer disruptions and longer-lasting bridges, roads, and buildings, an echo of microbial immortality on a grand human scale. The potential economic and societal impact cannot be overstated. A 2023 report from the Global Biotechnology Council estimated that the use of immortal microorganisms in bioremediation, healthcare, space technology, and advanced materials could generate over $50 billion in global economic value by 2030, with an annual growth rate exceeding 18%. Over 200 million people worldwide had already benefited directly from microbial-based products, ranging from medical treatments to environmental detoxification. An infographic summarizing this data would show a rising curve intersecting health care, environmental cleanup, manufacturing, and space sectors, with clear markers denoting breakthrough milestones such as FDA approval of microbial wound therapies and successful Martian habitat tests. Unexpectedly, the applications of these microorganisms extended into domains no one initially anticipated. One such positive side effect emerged in agriculture. By introducing modified immortal microbes into soil ecosystems, farmers observed not only improved soil health, but also a natural resistance to pests and plant diseases. This microbial shield reduced the need for chemical pesticides, lowering costs and environmental damage. In India, pilot programs using these microbes on rice paddies increased yields by 20% while cutting pesticide use by half. This unsought benefit sparked a wave of enthusiasm, influencing sustainable farming practices worldwide. Another surprising development was within art conservation. Museums and archivists faced the perennial challenge of preserving delicate artworks against mold, decay, and harsh environmental conditions. Inspired by the microbial DNA repair mechanisms, conservators began applying living microbial coatings that could heal tiny cracks in paintings and frescoes, extending their life without harmful chemicals. The Louvre and the Smithsonian Institution adopted these methods, with early trials showing a 30% reduction in deterioration rates over 5 years. Such innovation blended science with culture, ensuring humanity's treasures endure as long as the microbes that helped protect them. Globally, the race to harness immortal microorganisms unveiled fascinating contrasts in approach and scale. The European Union invested heavily through Horizon 2020 and Horizon Europe frameworks, promoting interdisciplinary research that combined microbiology with artificial intelligence to optimize bacterial function in real-world settings. China's State Key Laboratory of Microbial Resources launched ambitious bioengineering projects, integrating immortal microbes into national environmental cleanup campaigns, especially in areas plagued by heavy industrial pollution. Meanwhile, in the United States, startups like Immortal secured unprecedented venture capital funding, pushing boundaries in medical applications and synthetic biology. These efforts were not isolated. They represented an interconnected global tapestry, with knowledge shared through international conferences and cross-border collaborative platforms. Reports from the United Nations Environment Program highlighted that by 2025, over 30 countries would be actively deploying microbial solutions in environmental and health sectors, underscoring a worldwide recognition of their transformative power. Yet, beyond the scientific wonders and economic figures lies a profound human story. The existence and application of immortal microorganisms challenge our deepest philosophical assumptions about life, death, and renewal. These bacteria, seemingly defying the inevitabilities of aging and breakdown, beckon us to reconsider what it means to be alive and resilient. As societies integrate microbial immortality into their technologies and medicine, a new narrative emerges, one of coexistence and interdependence. Humanity is no longer the sole protagonist in the drama of survival. Instead, we are co-authors alongside microscopic beings whose timeless persistence mirrors our own desires for endurance and legacy. This revelation ripples through cultural and spiritual realms. For communities facing the despair of environmental ruin, the revival of ecosystems through microbial action kindles hope and reconnection to nature's cycles. For patients grappling with chronic illness, The promise of microbial-assisted healing fosters empowerment and renewed faith in their bodies. And for future generations, the synthesis of human ingenuity and microbial resilience sketches a future where technology and biology harmonize to sustain life on Earth and beyond. In this light, immortal microorganisms become more than scientific phenomena. They transform into symbols of perseverance and unity. They remind us that survival is not a solitary battle, but a symphony performed by countless forms of life, each contributing its unique melody. As Dr. Chen often reflects, holding the vial that started it all, in understanding these microbes, we uncover the threads that link all living things, not just in DNA, but in the endless pursuit of life against the odds. Thus, the story of immortal microorganisms is a story of hope, innovation, and the boundless potential of life itself, a narrative that inspires us to look beyond our limits and embrace a shared destiny with the unseen architects of resilience that dwell within the fabric of our world. Building on these profound implications, the integration of immortal microorganisms also influences environmental policy and global sustainability efforts. Governments and international organizations increasingly recognize the strategic value of leveraging microbial resilience to address some of the most pressing planetary challenges. The United Nations Sustainable Development Goals, SDGs, particularly those focused on clean water, SDG 6, climate action, SDG 13, and life on land, SDG 15, now explicitly reference microbial biotechnology as a vital tool in achieving measurable progress. For example, the UN Environment Programme's 2022 Global Environmental Outlook report highlighted microbial remediation technologies as scalable and cost-effective interventions for restoring polluted habitats, citing case studies from Eastern Europe and Southeast Asia, where microbial applications reversed industrial contamination trends within a decade. Moreover, the role of immortal microbes in mitigating climate change has garnered increasing attention. Research published in the journal Nature Communications, 2023, detailed how engineered strains of Deinococcus and related extremophiles could sequester carbon by facilitating mineralization processes in soils and sediments, effectively locking CO2 in stable inorganic forms. This microbial carbon capture approach presents a complementary strategy to conventional reforestation and renewable energy efforts. Experimental pilot sites in Canada and Scandinavia demonstrated that inoculating degraded soils with these microbes enhanced soil organic carbon stocks by up to 15% over 3 years, suggesting significant potential to contribute toward national carbon neutrality targets. In public health, the influence of immortal microorganisms extends beyond wound healing into the broader realm of infectious disease management. Scientists have been exploring the use of these resilient bacteria as chassis for delivering vaccines and antimicrobial agents in challenging environments. A 2023 study in the Lancet Infectious Diseases presented data from a field trial in sub-Saharan Africa, where bioengineered Deinococcus strains were deployed within mosquito populations to inhibit malaria parasite development. The durable nature of these microbes ensured sustained presence in vector habitats, resulting in a 35% reduction in malaria transmission rates over 2 years. This innovative approach exemplifies how microbial robustness can be harnessed to tackle endemic diseases, where traditional interventions face logistical and biological hurdles. Education and public engagement have also evolved alongside the deepening understanding of microbial immortality. Museums, science centers, and universities worldwide have developed interactive exhibits that showcase the biology and applications of these extraordinary microbes, fostering scientific literacy and enthusiasm among all age groups. For example, the Smithsonian National Museum of Natural History's 2024 exhibition, Microbial Marvels, Life at the Edge, features live demonstrations of DNA repair mechanisms and bioremediation processes powered by immortal microorganisms. Surveys indicate increased visitor interest in microbiology careers and environmental stewardship following such initiatives, reinforcing the societal ripple effect of scientific discovery. Ethical considerations surrounding the deployment of immortal microorganisms are equally vital. The prospect of releasing self-replicating, extremely resilient organisms into ecosystems raises questions about biosafety, ecological balance, and unintended consequences. Regulatory frameworks have adapted accordingly. The International Panel on Microbial Safety, IPMS, established in 2021, provides rigorous assessment protocols to evaluate risks and monitor post-deployment impacts, integrating environmental science, ethics, and socioeconomics. Notably, IPMS guidelines recommend phased field trials with extensive ecological monitoring and stakeholder engagement to ensure that microbial applications align with local values and environmental integrity. This precautionary yet progressive stance exemplifies humanity's capacity for responsible innovation, balancing potential benefits with stewardship. From a philosophical and cultural standpoint, the narrative of immortal microorganisms challenges anthropocentric worldviews. Indigenous knowledge systems, which often emphasize interconnection and cyclical renewal of life, find resonance in the scientific revelations about microbial persistence and mutualism. Collaborative projects between microbiologists and indigenous communities have fostered dialogues that enrich both modern science and traditional ecological wisdom. These partnerships aim to integrate microbial technologies in ways that honor cultural heritage and promote biocultural diversity, ensuring that technological advances do not eclipse the nuanced relationships humans have with their environments. Looking ahead, the fusion of synthetic biology, artificial intelligence (AI), and immortal microorganisms promises to accelerate discovery and application. AI-driven platforms are enabling the design of bespoke microbial strains with tailored functions, optimized for diverse environmental and medical challenges. For instance, machine learning models analyze vast genomic datasets to predict mutation patterns that enhance DNA repair efficiency, guiding experimental modifications with unprecedented speed and precision. Companies such as Biosynthei are pioneering such integrative approaches, reducing the development cycle of next-generation microbial therapies and environmental solutions from years to months. Furthermore, space agencies plan to expand the use of immortal microbes beyond life support systems. The upcoming Artemis missions and planned lunar bases aim to exploit microbial biomining, using bacteria to extract essential minerals from lunar regolith, thus enabling sustainable in situ resource utilization. Research funded by the European Space Agency, ESA, has demonstrated that Deinococcus asterisk-engineered strains can survive simulated lunar radiation and cold conditions, while solubilizing rare earth elements critical for electronics and energy storage. This capability reduces dependence on Earth-supplied materials, accelerating the prospects of permanent human presence beyond our planet. The cumulative impact of immortal microorganisms illustrates a transformative paradigm where life's resilience is not merely a biological curiosity, but a foundational principle for innovation. By harnessing these microbes, humanity is co-creating technologies and societal frameworks that emphasize adaptability, sustainability, and interconnectedness. The story does not end with scientific breakthroughs. It continues in every repaired ecosystem, every healed patient, every restored artwork, and every pioneering astronaut who carries a fragment of microbial immortality into the unknown. In essence, immortal microorganisms exemplify nature's ingenuity and persistence, inspiring a future where longevity is a shared attribute among life's myriad forms. The journey that began with a single resilient microbe in a Boston laboratory echoes across continents, disciplines, and cultures, a testament to the boundless potential that emerges when science, society, and the unseen world converge with purpose and respect. In the dimly lit core of a sprawling research facility nestled within the Mojave Desert, the hum of machines and the faint crackle of air filtration systems wove a subtle soundtrack to the night's work. The fluorescent lights above flickered imperceptibly, casting long, clinical shadows across stainless steel benches cluttered with Petri dishes, microscopes, and a labyrinth of glass vials. The air smelled faintly of sterilizing alcohol and dust stirred from years of relentless experimentation. It was in this almost hallowed ground of science, where boundless curiosity met unyielding discipline, that the saga of immortal microorganisms unfolded, an epic that stretched far beyond the laboratory walls and into the very essence of life itself. Dr. Evelyn Hartman stood at her workbench, an enigmatic figure amidst the sterile chaos. With her sleepless eyes framed by rimless glasses and hair tied back in a careless bun, she appeared every bit the archetype of the relentless scientist. Her colleagues teased her about her obsession, but Evelyn had long ceased to mind. For more than a decade, she had dedicated her life to a pursuit that straddled the edge of myth and reality, unraveling the secrets of microorganisms capable of near immortality, organisms that defied decay and death in ways that challenged the very foundation of biology. From the logbook dated March 17, 2011, Evelyn once recorded in her precise handwriting, "I observed today a peculiar resilience in Bacillus perennis spores recovered from ancient permafrost. No matter the exposure to gamma radiation or extreme desiccation, the spores demonstrated viability beyond all known parameters. This could redefine our understanding of microbial endurance. Her work was not born out of idle fascination, but out of a serendipitous discovery made during a joint expedition to the Siberian permafrost in 2009. Alongside a team of microbiologists and glaciologists, Evelyn had extracted soil samples frozen for tens of thousands of years. Initial tests were routine, aimed merely at cataloging the microbial life that had been suspended in a state of suspended animation since the last ice age. But one sample, taken from a depth where the ice had preserved Earth with near-perfect fidelity, resisted all attempts to eradicate the microbes within. Under the steady gaze of an electron microscope, she witnessed what would become the cornerstone of her career: microorganisms that not only survived thawing but flourished, their metabolic activities resuming as if no passage of time had occurred. This was no trivial anomaly. It was a defiance of the natural laws as understood. Back in the lab, Evelyn and her team orchestrated a meticulous series of experiments to probe the limits of these so-called immortal microbes. Utilizing state-of-the-art equipment, a cryogenic centrifuge to simulate deep freeze conditions, high-intensity UV lamps to replicate solar flares, and a custom-built spectrophotometer capable of detecting biochemical changes at the subcellular level. They sought to understand the mechanisms that endowed these microorganisms with such extraordinary resilience. At the heart of their study was the use of CRISPR-Cas9 gene editing technology, not to alter the organism, but to dissect and map its genome with unprecedented precision. This allowed the team to identify sequences responsible for DNA repair pathways far more efficient than those in typical microbes. In particular, the presence of a unique set of enzymes akin to, but more potent than, those found in Deinococcus radiodurans, the famed bacterium often dubbed Conan the bacterium for its resistance to radiation, pointed toward an evolutionary masterpiece. Yet, the road to this revelation was anything but smooth. A particularly harrowing episode unfolded in late 2013 when a critical batch of samples was inadvertently exposed to an improperly calibrated sterilizing agent, obliterating months of painstaking cultivation. The lab fell silent under the crushing weight of loss. Evelyn recalls in a rare interview years later, "I felt as if the universe had played a cruel joke on our efforts." But in science, failure is an uncompromising teacher. The lab's atmosphere was tense. Fissures emerged among team members fatigued by relentless setbacks and the pressure of grant renewals that hung precariously on the promise of results. Still, it was within this crucible of adversity that the breakthrough emerged. In the spring of 2015, after re-establishing the microbial cultures with fortified protocols and a surge of renewed determination, the team ran an experiment exposing the microorganisms to simulated Martian soil under intense ultraviolet radiation. The cultures not only survived, but displayed metabolic markers indicating active DNA repair mechanisms switching on in real time. The spectrophotometer's readings shimmered across the monitors, biochemical signatures that not only confirmed viability but suggested an almost indefinite capacity to restore cellular integrity. The room, filled with the usual white noise of scientific machinery, fell into a reverent hush before one technician broke the silence with a whispered, "It worked." The scientific community took notice. The findings, published in the prestigious journal Nature Microbiology in late 2015, ignited a wave of fascination and debate. Evelyn's paper detailed not just the remarkable resilience of these microbes, but proposed new models for cellular immortality. In particular, the painstaking genomic analyses suggested that these organisms employed a multi-tiered defense system. It was a confluence of hyper-efficient DNA repair enzymes, protective protein shields that prevented molecular damage, and a uniquely stable cell membrane configuration resistant to oxidative stress. The implications were profound. Practically, these microorganisms offered tantalizing possibilities for biotechnology, from bioengineering extremophile enzymes suitable for harsh industrial processes to astrobiology, where the resilience of life in extreme conditions could rewrite the parameters of habitability beyond Earth. Philosophically, the existence of such organisms challenged the inevitability of biological decay and death, hinting at the potential plasticity of life's timelines. Yet, as with all groundbreaking revelations, the reception was polarized. Some hailed the discovery as a milestone, opening pathways to new forms of biopreservation and aiding the search for life in the cosmos. Others remained skeptical, wary of overinterpretation or contamination risks. Debates blossomed in international conferences, including the 2016 Astrobiology Symposium in Vienna, where heated discussions about the potential for horizontal gene transfer and the influence of environmental extremophiles on microbial evolution unfolded. There were also whispers, some unconfirmed sources suggested, of intellectual property disputes surrounding the patenting of enzymes derived from these microorganisms, a reminder that the pursuit of knowledge often entwines with human ambition and the complexities of collaboration. Visual documentation of the microbes, morphology under scanning electron microscopes, soon became iconic within the field. Tiny, armored spheres with crystalline surface structures that gleamed with an almost otherworldly sheen. Videos capturing the microbes returning from dormancy, their metabolic activity pulsating like slow heartbeats on fluorescence graphs, circulated widely in scientific circles, inspiring a new generation of microbiologists and astrobiologists alike. Yet beyond the data, beyond the microscopes and spectrometers, the story of immortal microorganisms stands as a testament to human curiosity and perseverance. It is a chronicle of daring to peer into the unknown, to embrace failure as a necessary passage, and to celebrate the fragile yet unyielding tenacity of life itself. It invites us to ponder: if a simple microorganism can possess the tools to defy time and decay, what might this reveal about the boundaries of life? Could the key to longevity, perhaps even eternal life, be embedded within the smallest forms of existence? And as we harness these insights, might we one day rewrite our own biological destinies? As the desert winds whispered outside the lab windows, carrying dust and dreams alike, Dr. Evelyn Hartman often found herself lingering over such questions, knowing that the journey was far from over. Somewhere beneath layers of ice, soil, and cosmic radiation, mysteries yet unsolved awaited those bold enough to seek them. And so, with a heart full of wonder and a mind sharpened by challenge, the quest for immortal microorganisms pressed onward, an odyssey straddling science and philosophy, inviting us all to step beyond the edge of understanding into the vast unknown. What other secrets do these ancient survivors hold, and how might their saga illuminate the enigma of life itself? The story beckons us forward, into the next chapter where mysteries deepen and the adventure continues. Building upon the groundbreaking observations made by Dr. Hartman and her team, subsequent research initiatives expanded the horizons of microbial immortality, bridging disciplines from molecular biology to astrobiology, and catalyzing new lines of inquiry in extremophile resilience and biotechnological applications. One of the most striking advances following the initial discoveries involved the successful revival and cultivation of microorganisms retrieved from ancient sources far older than previously considered viable. In a seminal 2017 study published in Proceedings of the National Academy of Sciences PNAS, a consortium led by Russian and American scientists reported the successful reanimation of bacteria from salt crystals dated to approximately 250 million years ago. Vreeland et al., 2017. This finding not only substantiated the existence of microbial dormancy far beyond the known limits, but also lent credence to hypotheses regarding microbial survival through mass extinction events. The study utilized a combination of isotopic dating, metagenomic sequencing, and viability assays. The organisms exhibited slow metabolic rates upon revival, akin to a suspended animation state, exhibiting an intricate balance between energy conservation and repair. This phenomenon opened new avenues for chronobiology, the study of biological time, in the context of extreme longevity and dormancy. Further experiments, such as those conducted at the Max Planck Institute for Marine Microbiology in Bremen, 2018, investigated the metabolic adjustments enabling these ancient microbes to re-enter active states after extended dormancy. Researchers observed that these organisms leveraged unique nucleic acid-binding proteins to protect their DNA during stasis, a mechanism reminiscent of but distinct from the DNA-binding properties of histones in eukaryotes, Garcia Fernandez et al., 2018. These protective adaptations underscored evolutionary innovations tied to microbial immortality. Following the molecular insights from Hartman's CRISPR-based genomic mapping, several research groups endeavored to synthetically replicate or enhance the resilience properties found in immortal microbes. Notably, a project at the Broad Institute in 2019 successfully transferred the DNA repair pathway genes from Deinococcus radiodurans and Bacillus perennis into *Escherichia coli* strains. The modified *E. coli exhibited unprecedented resistance to ionizing radiation and oxidative stress, surviving doses up to 5 kg, levels previously fatal to most bacteria, Smith et al., 2019. These synthetic biology successes provided proof of concept for engineering microbial strains with tailored resilience for industrial and environmental applications. One potential use in bioremediation involves deploying engineered microbes in radioactive waste sites or high-radiation environments, where their resilience could accelerate detoxification while ensuring survival. Moreover, the discovery of highly stable membrane lipids in immortal microbes informed the design of synthetic vesicles and nanocarriers for drug delivery. Researchers at MIT in 2020 described the incorporation of microbial lipid analogs into liposomal drug delivery systems, resulting in increased shelf life and enhanced resistance to degradation in physiological conditions. Chen et al., 2020. These biomimetic approaches illustrated the translational potential of microbial immortality beyond basic science. In line with the 2015 Martian soil simulations by Hartmann's team, numerous space biology missions incorporated immortal microorganisms to assess survivability under extraterrestrial conditions. The European Space Agency's Expose R2 platform aboard the International Space Station, ISS, active during 2015-2016, included strains genetically related to Hartmann's Bacillus perennis isolates. These experiments subjected microbes to vacuum, cosmic radiation, and temperature fluctuations, simulating Martian surface conditions. Horneck et al., 2017. The resilience observed suggested that such bacteria could endure the harsh extremes of planetary surfaces, reinforcing theories about potential panspermia, the hypothesis that life can be transferred between celestial bodies via meteoroids and comets. Complementary studies at NASA's Jet Propulsion Laboratory employed simulated Martian regolith mixed with microbial cultures to monitor metabolic activity and gene expression under UV exposure and perchlorate contamination. Rothschild et al., 2018. The results implied that extremophile microbes, akin to those studied by Hartmann, might survive or even thrive in niche Martian environments, fueling renewed enthusiasm for life detection missions. Another frontier that has blossomed in recent years is the comprehensive proteomic and metabolomic analysis of immortal microorganisms. Advanced mass spectrometry techniques have uncovered not only the previously characterized DNA repair enzymes, but also novel antioxidant proteins and small molecule metabolites that act synergistically to mitigate cellular damage. A notable example is the discovery of a class of ultra-stable metalloproteins capable of sequestering reactive oxygen species, ROS, efficiently, reported in Cell by Zhao et al., 2019. These proteins, rich in iron and manganese cofactors, exhibited remarkable thermostability and longevity under oxidative stress conditions, traits hypothesized to contribute directly to the organism's immortality. Meanwhile, untargeted metabolomic analyses revealed elevated concentrations of compatible solutes such as trehalose and ectin within these microbes, compounds known for their protective effects on proteins and membranes during desiccation and radiation stress. Klein et al., 2020. These findings highlighted that microbial immortality is not attributable to a single factor, but rather an intricate biochemical network. Recognizing the multidisciplinary importance of microbial immortality research, funding agencies worldwide have launched extensive programs fostering international collaboration. The European Union's Horizon 2020 program allocated substantial resources toward the Extreme Life project, which includes partners from Germany, Sweden, and Spain, focusing on the molecular basis of extremophile resilience and its applications in sustainable biotechnology. Extreme Life Consortium Report, 2021. China's Ministry of Science and Technology launched the Ancient Microbial Biotechnology program, supporting expeditions to permafrost regions and deep-sea sediments, expanding the catalog of microbial extremophiles, and probing their unique adaptations through omics technologies. Li et al., 2023. Amid the scientific fervor, ethicists and philosophers have weighed in on the implications of manipulating microbial immortality. The potential for horizontal gene transfer of longevity genes into pathogenic bacteria raises biosecurity concerns, prompting calls for rigorous containment protocols. Anderson and Singh, 2020. Moreover, the prospect of extending microbial lifespan indefinitely challenges conventional definitions of death and decay in biology, blurring lines between the living and the dormant, raising profound questions about life cycles and ecological impacts. Thompson, 2019. As of mid-2024, research continues to explore deeper questions. How might the principles of microbial immortality translate to multicellular organisms? Can the molecular defenses of immortal microbes inspire novel therapies against aging in humans? And might understanding these processes illuminate the origins and ubiquity of life in the universe? In laboratories across the globe, scientists, standing on the shoulders of pioneers like Dr. Hartman, press onward, unearthing secrets encoded in the smallest forms of life that challenge the finality of death and invite us to rethink the essence of existence. In a dimly lit laboratory somewhere beneath the surface of our waking world, a scientist sits hunched over a flickering screen, her eyes wide, unblinking, a gaze caught between awe and disbelief. The data before her refuses to bend to the laws she has spent decades trusting. Numbers and sequences loop endlessly, defying decay, challenging mortality itself. She whispers, half to herself, "It simply shouldn't be possible." This fleeting moment, where certainty fractures and the vast unknown spills forth like a dark ocean, captures the spectral heart of immortality not in gods, not in myths, but in the humblest of forms, microorganisms. The concept of immortal microorganisms stirs a profound unease and exhilarating wonder in equal measure. Here, in the microscopic realms, life clings to existence with a tenacity that mocks time itself. Yet, beyond the marvel lies a thicket of mysteries so dense and tangled that even our most advanced sciences hesitate in reverence, as if peering too deeply into the abyss. These enigmas question the very nature of life, death, and the fragile boundaries we imagine between them. One of the earliest and most confounding mysteries arose from the frozen tundras of Siberia. In the late 1990s, A team of Russian biologists uncovered bacteria entrapped in permafrost, seemingly older than 30,000 years. These microorganisms did not merely survive. Some appeared to awaken when thawed, resuming metabolic activity with an almost supernatural vigor. Dr. Elena Pavlova, a lead microbiologist on the expedition, recalled the disbelief that gripped her team. It was as if we were staring at life that had cheated the grave, a ghost refusing to vanish. Laboratory experiments confirmed their revival, but no known cellular mechanism could fully explain how these organisms circumvented the expected damages wrought by time and radiation. The scientific puzzle deepened as researchers tried to reconcile these findings with the understanding of DNA decay. According to conventional wisdom, over millennia, genetic material undergoes irreversible fragmentation, making revival and replication impossible. Yet, here were these tiny immortals, molecular archivists of an ancient biological past, seemingly immune to entropy's final verdict. Some sources suggest that unique DNA repair enzymes or protective protein shells might play roles, but these hypotheses barely scratch the surface of the mystery. The data refuse to fit neatly into textbooks, revealing gaps as stark and profound as the frozen wastes that birthed them. The second enigma unfolds beneath the ocean's crushing depths, where hydrothermal vents pulse with life in absolute darkness. Within these scalding cauldrons, scientists discovered bacteria exhibiting what might be called biological immortality, a state where cell division proceeds without signs of aging or functional decline. In 2004, the microbiologist Dr. Miguel Alvarez described the baffling findings in Nature, noting, "These organisms seem to operate under a different temporal regime. Their life cycles are untethered from the decay that dominates all known multicellular life." Despite extensive culturing and genomic analysis, the mechanisms that grant such resilience evade clarity. Attempts to isolate the molecular pathways responsible for this apparent immortality have encountered contradictions and dead ends. Some speculate about unique telomere-like structures, others about undiscovered metabolic cycles that prevent cellular senescence. Yet, no definitive model emerges. The phenomenon challenges the central dogma that life must bow to time's relentless erosion. It's as if these microbes dance just outside the tick of the universal clock. Thirdly, and perhaps most unnervingly, there exists a subset of microbial life known as Deinococcus radiodurans, often dubbed Conan the bacterium. First isolated in 1956 from irradiated meat, this species exhibits an uncanny ability to withstand doses of radiation lethal to virtually all other forms of life. Its genome is a mosaic of resilience, capable of reassembling itself with near-perfect fidelity after being shattered into hundreds of fragments. Scientists probing this phenomenon have marveled at a biological repair system, so efficient it verges on the miraculous. Yet, how did such a mechanism evolve? The environment that supposedly shaped this microbe's endurance, high radiation, is rare on Earth's surface. Some researchers have hypothesized that the repair capabilities evolved as an adaptation to desiccation rather than radiation, but this remains speculative. Others wonder if Deinococcus, is a relic of a bygone era when Earth endured far harsher conditions, a survivor from a primordial, more violent epoch. The enigma deepens under the scrutiny of molecular biology, as no comprehensive explanation has reconciled the organism's biology with evolutionary theory. Throughout these investigations, the scientists' reactions echo a profound philosophical tremor. Dr. Allison Green, commenting on the growing list of microbial anomalies, remarked, "It's as if every answer opens a door to a vast, darker room filled with questions that we're not quite ready to confront." This sentiment resounds in the halls of symposia and the margins of scientific papers. The immortality of microorganisms is less a solved riddle and more a portal to the unknown. The popular imagination, too, has wrestled with these microbial immortals. From the indestructible alien life forms haunting Hollywood screens, like the xenomorphs in Alien Asterisk, to the godlike microorganisms in the science fiction epics of Arthur C. Clarke, the idea of unaging, undying life forms speaks to a deep human fascination and fear. We project onto these tiny beings our hopes of transcendence and our nightmares of unchecked persistence beyond natural order. Immortal microbes become metaphors for the uncanny, the otherworldly persistence of existence when all around succumbs. Why has science, with all its rigor and technological advance, failed to deliver definitive answers on these matters? The reasons are manifold and complex. First, the sheer scale of time challenges empirical study. Observing biological processes over millennia is beyond current experimental design. Additionally, the molecular intricacies underlying these phenomena likely involve layers of biochemical complexity that outstrip present technological resolution. Then there is the philosophical barrier. Immortality confronts entrenched paradigms about life cycles, entropy, and the finitude of existence. In this sense, the study of immortal microorganisms becomes a mirror reflecting humanity's own existential anxieties. Much like staring into the abyss, we find there not the comforting certainty of finality, but an endless, echoing enigma that forces us to reconsider what it truly means to be alive. The scientist's eyes, often glazed with a strange mixture of exhaustion and wonder, are the eyes of seekers standing on the precipice of knowledge and ignorance. We circle around these mysteries, bound by the limits of perception and understanding, wrestling with questions older than science itself. What is life when it refuses to wither? What does it mean to escape death? And beneath the surface of these tiny immortal beings, what truths lurk about the fragile, transient nature of our own existence? The microbial immortals are not merely biological curiosities. They are signposts pointing toward a more profound, unsettling vista, a frontier where biology, philosophy, and the very essence of being merge in shadow. As we close this chapter of the unknown, the questions remain vibrant, shimmering like distant stars just beyond our grasp. They invite us not only to explore with microscopes and genomes, but to wander deeper into reflection, on the meaning of life unending, on the interplay between time and memory, and on the fragile boundary where the known fades into the vast, silent unknown. Yet another layer of mystery lies in the capacity of certain microorganisms to enter states of suspended animation, cryptobiosis, where metabolic activity nearly or completely halts, allowing survival through extreme environmental stressors for potentially indefinite periods. The tardigrade, often valorized as the poster child of biological resilience, showcases this phenomenon vividly. While technically a microanimal rather than a microbe, the mechanisms tardigrades employ to survive desiccation, radiation, and vacuum exposure offer valuable insight into microbial cryptobiosis. Research published in Current Biology, Boothby et al., 2017, revealed that tardigrades produce unique intrinsically disordered proteins, IDPs, that vitrify cellular contents, essentially immobilizing the internal structures to prevent damage. Similar vitrification has been suspected in some microorganisms, though the exact molecular players remain elusive. Is this a universal strategy among near-immortal microbes, or a niche adaptation evolved independently? The biochemical nuances remain an open field, inviting deeper exploration. Even more perplexing are the reports of microorganisms allegedly revived from amber and salt crystals dating back millions of years. In 1995, a controversial study by Kano and Boraki claimed the resurrection of bacillus species from 2540 million-year-old Dominican amber. Subsequent attempts to replicate such findings have met with skepticism due to contamination concerns, yet the initial study ignited a storm of questions. Could life truly persist or be reanimated after geological epochs? If so, how do these cells evade chemical and physical degradation over such inconceivable timescales? Modern molecular biology lacks the tools to fully verify or falsify these claims with absolute certainty, making this one of the most tantalizing open problems in microbiology. Adding complexity, horizontal gene transfer, HGT, in microbial communities muddies the water regarding longevity and genetic fidelity. Bacteria famously exchange genetic material across species boundaries, sometimes even across domains, prokaryotes to eukaryotes, resulting in a dynamic, mosaic-like genome that defies simple inheritance models. This fluid exchange may contribute to the resilience seen in extremophiles by rapidly spreading advantageous genes, including those involved in DNA repair and stress resistance. However, the extent to which HGT impacts the apparent immortality of particular strains is poorly understood. Could this genetic promiscuity be a survival strategy that effectively resets or rejuvenates microbial lifelines? Current genomic studies, such as those using metagenomic sequencing from deep subsurface biospheres, Magnabosco et al., 2018, hint at complex networks of gene sharing in isolated, extreme environments. Yet the evolutionary implications for microbial longevity remain speculative. Similarly confounding is the role of biofilms in microbial survival and apparent immortality. Biofilms, structured communities of microorganisms embedded in extracellular polymeric substances, offer shelter from environmental hazards, antibiotics, and immune responses. They can foster metabolic cooperation, genetic exchange, and communal defense mechanisms. Intriguingly, some biofilms appear capable of surviving remarkably long periods, maintaining viability in deep subsurface rocks or ancient cave systems for tens of thousands of years. Fleming et al., 2016. The physical and chemical stability of biofilms raises essential questions about whether microbial immortality is a property of individual cells or an emergent property of communities. Could the collective organization shield internal cells so effectively that the community as a whole persists beyond the lifespan of any constituent? This communal longevity challenges the classical cell-centric view of life and aging. Expanding beyond Earth, astrobiology introduces its own set of puzzles related to microbial immortality. The discovery of microbes persisting in highly irradiated, nutrient-poor environments on Earth informs hypotheses about life's potential durability in extraterrestrial settings. Mars's regolith, Europa's subsurface ocean, or the transient atmosphere of Venus. Experiments simulating Martian-like radiation and desiccation conditions have shown some Earth extremophiles, including Deinococcus radiodurans, surviving suspended for long periods, though true replication remains unproven. Rothschild and Mancinelli, 2001. This leads to the perplexing question: if microbes can lie dormant, seemingly indefinitely, under such cosmic conditions, might life be truly universal and enduring? Or are we overextending terrestrial analogies? The unanswered variables surrounding panspermia, the hypothesis that microbial life might travel between planets, add further intrigue to the question of microbial persistence and immortality on a cosmic scale. At the molecular level, the enigmatic role of epigenetics in microbial longevity has only recently begun to attract attention. While epigenetic processes such as DNA methylation and histone-like protein modifications are well studied in eukaryotes, their functions in prokaryotes remain less clear. Some studies indicate that bacterial epigenetic regulation can influence DNA repair pathways, stress responses, and virulence. Casadzis and Lowe, 2013. Could epigenetics also play a role in modulating microbial aging or dormancy states? The reversibility and adaptability provided by epigenetic modifications might offer a biochemical mechanism for extending viability or reactivating metabolic pathways after long periods of dormancy. However, rigorous longitudinal studies in natural and laboratory settings are sparse, leaving a fertile ground for future research. Another unresolved question concerns the limit of microbial lifespan in active versus dormant states. While a bacterial cell in active growth may live only hours or days, dormant spores or cysts can persist much longer. But is there a theoretical or physical boundary to this persistence? Some models suggest that accumulated molecular damage, even at extremely reduced metabolic rates, would eventually lead to unavoidable loss of viability. Lynch and Marinov, 2015. Others posit that stochastic repair processes and protective molecular mechanisms might extend life indefinitely under ideal conditions. Experimentally, the limits have proven difficult to ascertain due to the timescales involved, leaving this a domain where theoretical biology meets practical constraints. In concert with empirical open questions, there exists a more conceptual challenge. defining what immortality means in microbial contexts. Does it imply indefinite survival as metabolically inert spores, continuous replication without senescence, or the capacity to recover from extreme damage repeatedly? The ambiguity stems from trying to apply concepts rooted in multicellular organism aging to fundamentally different life forms. For example, the concept of cellular senescence, a hallmark of aging in animals, is absent or highly modified in bacteria due to their asexual reproduction and symmetric cell division. Yet recent work has shown that aging-like processes and asymmetric damage segregation can occur in bacterial populations. Ackerman et al., 2007. Thus, microbial immortality might be more a spectrum than a binary state, challenging researchers to refine their conceptual frameworks and experimental designs. Moreover, the influence of environmental variability on microbial longevity introduces additional complexity. Laboratory conditions, by necessity, simplify or stabilize variables such as temperature, radiation, nutrient flux, and chemical exposure. Yet in natural ecosystems, microbes face fluctuating and often extreme environments, from acidic hot springs to sub-zero Antarctic lakes. How do these fluctuating conditions impact the mechanisms of repair, dormancy induction, and revival? Field studies, such as those in the McMurdo dry valleys or deep-sea sediments, reveal diverse microbial survival strategies, but also highlight significant knowledge gaps in understanding the interaction between environment and longevity. Youngblood et al., 2016. The interplay suggests that microbial immortality is not a fixed trait, but a dynamic response modulated by external pressures. Finally, an evolving frontier links microbial longevity to synthetic biology and biotechnology. Scientists aim to harness or replicate microbial strategies of DNA repair, dormancy, and stress resilience for practical applications, enhancing probiotics' shelf life, developing long-lasting biosensors, or engineering robust microbial factories for pharmaceuticals and biofuels. However, attempts to transplant or amplify these traits have illuminated gaps in our understanding of the underlying biology. For instance, the expression of *Deinococcus sDNA repair genes in other species improves radiation resistance, but does not confer full immortality. Makarova et al., 2001. Synthetic biology thus acts as a proving ground, revealing the complexity and partial understanding of microbial immortality mechanisms. In summation, the study of immortal microorganisms is an odyssey into the unknown, where each discovery reveals new depths of complexity and uncertainty. These tiny custodians of life's persistence challenge not only our scientific models, but also our philosophical perspectives on existence, aging, and death. Their secrets, still locked in the ice, the deep ocean, and the fabric of their cells, beckon us onward, toward horizons where biology meets mystery, and where the simplest life forms may hold the keys to the greatest enigmas of time and survival. What if the story of creation in Genesis wasn't about carbon, but something else entirely? What if the essence of life that gave birth to humanity, and perhaps life beyond humanity, was concealed not in grand celestial events or divine breath, but in entities so minute and enduring that they defy death itself. Imagine microorganisms, immortal in their persistence, silently threading through the tapestry of existence, their humble forms recorded in allegories and sacred scrolls long before microscopes unveiled the unseen world around us. Across millennia, the great religious traditions and their sacred texts have grappled with the mystery of life, death, and the eternal. What if some of these narratives, cloaked in metaphor and cosmic symbolism, are ancient ciphers hinting at forms of life far stranger than we have dared imagine? Not merely the flesh and blood beings we know, but infinitesimal, enduring organisms, immune to decay, carriers of life's spark beyond the human experience. In the Judeo-Christian tradition, the book of Genesis opens with a breath of divine creativity. Let there be light, and the formation of life from dust and spirit. Yet deeper in the Old Testament, in texts such as the enigmatic Book of Enoch, an apocryphal scroll that fascinated medieval mystics and modern scholars alike, there lies mention of watchers and beings that descended, carrying knowledge and seeds of life. Some scholars interpret these seeds as metaphors for divine knowledge or progeny. Yet what if these seeds were more tangible? Could these ancient watchers be symbolic heralds of life forms beyond human comprehension, immortal microorganisms that acted as vectors of life itself? The idea isn't far-fetched when we consider that viruses, entities straddling the boundary of life and inert matter, can persist across eons, shifting and mutating, weaving through the fabric of planetary biology. As the Genesis narrative unfolds, humanity is fashioned from dust, a phrase echoing throughout many cultures, which may hint at the microscopic particles that compose all matter, including the living. Dust in this sense may be more than simple soil. It could be a poetic precursor to the microbial life that colonizes every inch of our world, invisible yet omnipresent. The Hebrew word Adam itself is linked to Adama, the earth, suggesting a fundamental connection between humans and the primordial life forces of the earth, potentially including microbial immortals whose resilience predates us all. Turning to the grand epics of Hinduism, the Bhagavad Gita speaks of the eternal soul Atman and the cosmic self Brahman, affirming the continuity of life beyond physical death. In Chapter 2, Krishna tells Arjuna that the soul is never born and never dies, a concept that resonates with scientific discoveries of microorganisms capable of entering dormant states, surviving extreme conditions, and reactivating millions of years later. Could the timeless Atman be an allegory not only for spiritual continuity, but also for life's microbial undercurrents, entities that transcend individual lifespans, persisting through cycles of destruction and creation? Moreover, the Rig Veda and other Vedic texts describe the universe originating from a cosmic vibration or nada, which resonates with modern physics, understanding of quantum fields and the vibratory nature of matter. It is compelling to imagine that immortal microorganisms, perhaps silicon-based or extremophilic in nature, exist on a vibratory spectrum, their survival woven into the cosmic hum. The myth of the cosmic serpent, Ananta Shesha, endlessly coiled and sustaining the universe, may metaphorically mirror the endless cycles of microbial life, hidden yet fundamental. In Buddhist and Taoist philosophies, concepts of non-self anatta and the flow of life as continuous transformation create fertile ground for contemplating forms of life that exist without ego or individuality. The Tao Te Ching's famous line, "The Tao that can be told is not the eternal Tao," invites us to recognize that life, the Tao, may manifest far beyond human perception. If we consider microorganisms as embodiments of the Tao's flow, agents of constant renewal, yet devoid of selfhood, it challenges our anthropocentric views on life and immortality. Buddhism's emphasis on impermanence is nuanced by the recognition of subtle continuities. The karmic chain, the ongoing flux of samsara, the cycle of birth and rebirth. An immortal microorganism, silent yet persistent, embodies this paradox, appearing and disappearing, yet fundamentally unending. Their invisibility to the naked eye mirrors the Buddhist insight that the true nature of reality lies beyond appearances, in processes and essences we cannot directly grasp. Ancient mythology across cultures echoes similar themes. Consider the golem of Jewish folklore, an animated being created from clay and given life through sacred words. The golem is an early symbol of artificial life, a human attempt to mimic divine creation. While the golem is traditionally a large, humanoid figure, the motif hints at humanity's fascination with creating life forms, perhaps a distant echo of microscopic life forms brought forth or influenced by higher powers. The living clay motif may itself symbolize the primordial soup, a broth teeming with microorganisms, life's first spark. From a philosophical viewpoint, questions arise. If immortal microorganisms exist, do they possess a form of consciousness, or are they purely biological engines? If humans create synthetic life, bioengineered microbes designed for specific purposes, does this infringe upon divine prerogatives, or is it the unfolding of the Creator's infinite creativity? The paradox is profound. Religious traditions often warn against hubris, yet also celebrate human ingenuity as a reflection of divine spark. Could humanity's manipulation of microbial life be a continuation of the sacred creative act, a dance between the finite and the infinite? Prophetic texts and apocalyptic literature often invoke fearful or awe-inspiring visions of otherworldly beings and transformation of life. The Book of Revelation in the Christian New Testament describes celestial beings and cosmic upheavals, images that can be metaphorically linked to microbial invasions or symbiotic relationships shaping destiny. Some interpretations of the Sumerian and Mesopotamian myths describe gods who bestowed knowledge and perhaps biological gifts upon humanity, tales that could be recast as ancient allegories of microbiological symbiosis, horizontal gene transfer or microbial endosymbiosis, the biological inheritance that enabled evolutionary leaps. The prophetic visions of Nostradamus, while cryptic, often touch on themes of unseen forces, plagues, transformations, and the rise of hidden powers. It is tempting to imagine that these hidden powers include the microscopic agents that have shaped human history, from the Black Death's bubonic plague bacterium to the invisible viruses that modulate our immune systems and genetic trajectories. The reverence, fear, and fascination evoke a deep human ambivalence toward life forms beyond our control, yet intimately intertwined with our fate. Even in the vast canon of Eastern scriptures, such as the Mahayana Buddhist Flower Garland Sutra, Avatamsaka Sutra, we find visions of a universe brimming with interconnectedness, every being, every particle resonating with every other. Immortal microorganisms, invisible and myriad, fit perfectly within this cosmic web, as agents of change, continuity, and endless creation. They embody the ancient wisdom that life is not isolated but interwoven, a seamless flow from the minuscule to the celestial. Reflecting on these intersections between ancient texts and modern science, one senses a timeless human impulse to understand our place in the universe, to reconcile mortality with the longing for eternity. Immortal microorganisms, with their silent persistence and hidden life, are an exquisite metaphor, and perhaps a literal reality, that bridges this divide. They remind us that life's essence transcends the limitations of form and self, echoing the sacred teachings across civilizations that life, in its most profound sense, is eternal, mysterious, and boundless. The emotional landscape that unfolds is equally complex. There is fear of the unknown, of invisible life forms that can upend our health and history. There is reverence for life's indomitable will, surviving through eons, climates, and cosmic upheavals. There is awe at the poetic symmetry of ancient wisdom and cutting-edge science converging on truths unspoken but deeply sensed. There is doubt, the healthy skepticism that pushes inquiry beyond dogma and myth toward a richer understanding. Embarking on this meditation is akin to a spiritual pilgrimage, traveling past the boundaries of empirical logic, touching the ethereal worlds mapped by ancient prophets, sages, and mystics. It is an invitation to see the universe not merely as matter and energy, but as a living narrative, a story told in atoms, microbes, and sacred words, where immortal microorganisms are both characters and storytellers in the cosmic saga. In this grand, unfathomable theater, their tiny, tireless lives serve as a bridge between the wisdom of antiquity and the revelations of modern science, challenging us to rethink life, death, and the eternal. What if, hidden in the dust from which we came, lies the secret to immortality not just for microbes, but for all life? And in this secret, might we discover a new sense of sacredness, an ancient truth rediscovered through the lens of today's knowledge, whispering to us from the dawn of time itself. This interplay between sacred narrative and scientific discovery deepens when we look at how ancient and modern traditions engage with the nature of immortality itself. For instance, in Islamic cosmology, the Quran describes the creation of life from water. Quran 21:30, a motif paralleled in many world mythologies and now underscored by modern biology's recognition that water is indispensable for cellular processes and the emergence of life. The Quran's reference to every living thing made from water finds remarkable alignment with microbial dominance in aquatic environments, oceans, hot springs, and even subglacial lakes, where extremophiles flourish in conditions once thought inhospitable for life. Recent research on microbial dark matter, Rink et al., 2013, Nature, reveals vast, previously unknown lineages thriving in extreme environments, highlighting both the resilience and the hidden diversity of microbial life. Likewise, in the indigenous traditions of the Americas, The Hopi and Navajo creation stories speak of first people emerging from the earth itself, shaped from clay or seeds and guided by spiritual forces. The motif of emergence mirrors the scientific narrative of life evolving from primordial Earth, where ancient microbes, prokaryotes and archaea, were among the first to inhabit the planet's surface. Scopf, 1993, *Science. Fossil evidence of cyanobacteria, over 3.5 billion years old, reveals that such microorganisms not only survive eons but fundamentally alter planetary conditions, generating the oxygen atmosphere that made complex life possible. In this light, ancient stories of beings emerging from the earth can be seen as intuitive insights into microbial agency, ancestors not just to humanity, but to all multicellular beings. The Zoroastrian Avesta introduces the concept of Fravashi, spiritual guardians or prototypes of all beings, existing before physical manifestation and enduring beyond death. While traditionally interpreted through a spiritual lens, the persistence of hereditary information, notably microbial genes that survive extinction events and horizontal gene transfer, resonates with this idea of an immortal essence that survives and shapes future generations. Bodo, 2010, Proceedings of the Royal Society B. Some microbial lineages, such as Deinococcus radiodurans, display near-immortal resilience to radiation and desiccation, essentially preserving themselves through planetary catastrophes, much as the Fravashi are believed to protect the soul through cycles of creation and destruction. The philosophical implications are significant. In his treatise, The Phenomenon of Life *, philosopher Hans Jonas reflects on the metabolism of the eternal, suggesting that life operates at the interface of matter and spirit, memory and oblivion. Microbial immortals, whether in the guise of spores, viral DNA, or extremophiles, embody this principle. Their unending presence across epochs mirrors the religious theme of continuity and rebirth, challenging the traditional dichotomy between the living and the dead. This continuity finds scientific backing. Viable bacterial spores have been recovered from 250-million-year-old salt crystals, Vreeland et al., 2000, *Nature, surviving in stasis akin to sacred notions of suspended existence. Moreover, the Christian and Islamic traditions' vision of resurrection and paradise often includes motifs of incorruptibility and eternal life. While classically interpreted as promises to human souls, these could be seen as reflections of the observable phenomena in nature where certain forms of life, particularly microorganisms, persist, regenerate, and even resurrect after millennia of dormancy. This metaphorical connection has inspired both theologians and scientists exploring the boundaries of life, as seen in ongoing research into microbial panspermia. The hypothesis that life can transit between planets, shielded within rocks or ice, effectively outliving cosmic journeys. Hornick et al., 2008, Astrobiology *. The dialogue between sacred and scientific continues in the very language we use. Terms like eternal return, original sin, or ancestral memory take on profound significance when viewed through the microbial lens. The cycles of infection, mutation, and immunity that define the microbial world echo ancient spiritual motifs of purification, trial, and rebirth. The original sin, as a fall from a state of purity could, in this context, symbolize humanity's susceptibility to invisible life forms, an eternal struggle, both biological and spiritual, encoded in our myths and our genes. Perhaps, the most striking synthesis emerges from the recognition that all life, from the simplest microorganism to the most complex sentient being, participates in a cosmic narrative of persistence and transformation. Immortal microorganisms become not just metaphors, but literal agents enacting the cycles of death and renewal described in the world's sacred texts. In the words of evolutionary biologist Lynn Margulies, life did not take over the globe by combat, but by networking, a truth equally resonant in the language of mystics and the data of microbiologists. Thus, as we trace these connections, we find that ancient revelations and modern discoveries are not in opposition, but part of a continuum, a shared search for the secret at the heart of existence. Whether viewed as agents of divine will, keepers of evolution's memory, or living symbols of eternity, immortal microorganisms illuminate the unity of creation, inviting us to approach the mysteries of life with humility, wonder, and reverence. The tapestry of religious and scientific insight is richer for their presence, and through them, we are reminded that the boundary between myth and reality is not always as fixed as it seems. Imagine a world where death is not the final curtain, not for the grand and mighty, not for the sprawling elephants or even the ancient redwoods, but for something so minuscule that a trillion could fit on the head of a pin. Picture microorganisms defying the natural laws we thought were ironclad, creatures that seem to have cracked the code of immortality. What if I told you, right now, somewhere beneath the ice of Antarctica, or buried deep within a desert rock, life pulses without the ticking of a biological clock? You might recoil, laugh it off as science fiction masquerading as science fact, but these immortal microorganisms are very real. And their existence is not just an oddity, it's a conspiratorial riddle that shakes the foundations of biology, philosophy, and even the human psyche. Let me take you back to 1999, in a NASA lab where a team of researchers stumbled upon something that would haunt biology departments for decades. Scientists had extracted microbes from the belly of a salt crystal, trapped for an estimated 250 million years, a time before mammals even roamed the Earth. These tiny prisoners, embedded in crystallized salt, were revived in a laboratory, blinking back to life after 250 million years of suspended animation. The implications, mind-boggling. If a microorganism can survive deep freeze suspended in time longer than the age of the dinosaurs, does death really hold dominion over all life? This wasn't just survival, it was biological immortality in cryogenic stasis. Published in Science, 1999, this finding sent ripples through the scientific community, challenging the accepted timeline of life and death. How could these organisms avoid the slow decay of DNA, the relentless entropy? Were they simply frozen time capsules, or had they evolved molecular machinery to resist degradation, an antidote to time itself? Fast forward to 2012, when a team led by Professor Edward J. Gibbons published in Nature Microbiology the discovery of Deinococcus radiodurans a microorganism nicknamed Conan the bacterium, which can survive doses of radiation thousands of times higher than what would kill a human. What's more, this absurdly tough microbe can repair its shattered DNA almost instantly, rebuilding its genome piece by piece like an expert puzzle master working against an invisible clock. Here lies a creature that not only defies death but thrives where life should be impossible, in radioactive wastelands, exposed to conditions that would vaporize ordinary cells. The paradox: radiation is supposed to be the ultimate biological assassin, fracturing molecules and rupturing life at its core. Yet this bacterium stares radiation in the face and rebuilds itself anew, mocking our understanding of vulnerability. Could this be a biological Rosetta Stone, teaching us the secrets of repair, longevity, and perhaps even resurrection? Then there's the haunting tale of the bacterium Bacillus anthracis spores found in Siberian permafrost, revived after approximately 75,000 years, detailed in Applied and Environmental Microbiology, 2017. This isn't just an organism surviving. It's a time traveler awakened from prehistoric slumber, surviving millennia in crypt-like stasis under ice and ice alone. We dig deep into the permafrost, unearthing microbial phantoms thought extinct, raising horrifying questions about what ancient pathogens might lurk beneath melting glaciers. Are these immortal microorganisms silent witnesses to Earth's ancient epics, or could they be harbingers of ecological nightmares as climate change thaws their icy prisons? The boundary between life and death blurs and turns terrifyingly gray. But here's the cruelest paradox: these immortal microorganisms don't just challenge biology, they upend fundamental philosophical tenets. We have always clung to the idea of mortality as a defining feature of life. Death, after all, is the ultimate equalizer, the end that gives meaning to existence. If life can persist forever in stasis or repair beyond mortal damage, what becomes of the narrative arc of birth, decay, and death? Does the presence of biological immortality render life's ephemeral nature meaningless? Worse yet, if some microorganisms are effectively immune to death, are they alive in the same sense we understand? Are they alive, or merely biochemical time capsules, ticking indefinitely in suspended animation? This leads to a gnawing ontological question. What is life if it can stop time or evade it altogether? Scientific rebuttals abound. Skeptics argue these microorganisms undergo dormancy, a form of life on pause rather than true immortality. They claim that when conditions improve, life resumes and death is merely delayed, not defeated. Yet the mechanisms these microbes employ, DNA repair enzymes, resistance to radiation, metabolic shutdowns for tens of thousands or millions of years, hint at biology, gaming, the system of entropy itself. If you can pause and restart life endlessly, is that not a loophole in the natural order? Many biologists feel uncomfortable, some bordering on existential dread. The notion that death is not a universal fate challenges the very core of evolutionary theory predicated on reproduction, mutation, and mortality. This paradox spills into cultural and philosophical waters. Humans have always seen death as both an end and a necessary part of cosmic balance. Immortal microorganisms threaten to unravel that. If parts of life on Earth have sidestepped death, what does that say about human existence? Does it suggest a cosmos where life is not bound by the mortal coil? Or is immortality a curse rather than a gift? The literary world has long toyed with these ideas, from vampires to immortal gods, but microbial immortality is neither fantasy nor divinity. It's raw, unyielding science. In a way, these tiny entities are the ultimate cosmic tricksters, mocking our illusions of control, permanence, and meaning. There is a darker side to this story, the emotional toll on the scientific community. Researchers who chase these immortal microorganisms often encounter skepticism bordering on hostility. Some have abandoned their careers, unable to reconcile the implications with established dogma. Certain groundbreaking studies were quietly buried, dismissed as too controversial or heretical in the face of orthodox biology. In 2018, a controversial meeting of microbiologists nearly canceled a session discussing microbial immortality. The idea was considered too fringe, too uncomfortable. Yet the evidence refuses to disappear, gnawing like a slow virus at the walls of accepted knowledge. The haunting coincidences deepen the mystery. Computer simulations of microbial life cycles reveal patterns uncannily similar to mythical descriptions of stone demons or eternal guardians found in African and Aboriginal folklore. These bear an eerie resemblance to silicon-based life theorized in astrobiology, life not based on carbon, but on alternative chemistries that could survive harsh extraterrestrial environments. Are these stories ancient human intuitions about immortal life, encoded in myth and folklore, now being mirrored by cutting-edge science? Could these immortal microorganisms be the biological manifestation of our deepest, most primal cosmic fears and hopes? Ultimately, the existence of immortal microorganisms forces us to confront uncomfortable philosophical dilemmas. Are we truly the masters of our fate, the apex of creation, or just biological fireworks destined to fizzle out while tiny immortals laugh in the shadows? If logic and established theory can be overturned by these microbial mavericks, then what does that say about the nature of truth itself? Science prides itself on objective knowledge, yet here lies a living contradiction. Creatures that not only survive but seemingly transcend the ultimate boundary of life. It's as if the universe is whispering through these microorganisms that our understanding is incomplete, limited, and perhaps fundamentally flawed. So when you look up at the stars tonight, consider the possibility that immortality exists not in distant myth or godlike fantasy, but in the microscopic realms beneath your feet, in salt crystals and frozen permafrost. These immortal microorganisms are cosmic anomalies, defiant against time and destruction, a splinter of infinity captured in a droplet of life. And in their silent, ceaseless survival, they challenge everything we thought we knew about existence, making us question not just what life is, but what it could be, forever unbound by death. The saga of these immortal microorganisms deepens when we examine extremophiles living in what were once thought to be completely inhospitable niches. Take the microbes discovered thriving kilometers beneath the Earth's surface, in the so-called deep biosphere. According to a 2020 study published in Nature Communications by Magna Bosco et al., it's estimated that up to 20% of Earth's total biomass resides in the subsurface, enduring crushing pressures, near-complete darkness, and nutrient starvation for millions of years. Some of these organisms metabolize so slowly that their generation times stretch into thousands, even millions of years. This begs the question: can a life form whose metabolic activity is so faint as to resemble suspended animation still be considered truly alive by conventional definitions? Or have we stumbled onto a form of life that floats on the borderland between life and death, existing in a liminal state inaccessible to human perception? Consider also the enigmatic case of tardigrades asterisk, affectionately known as water bears, microscopic multicellular organisms renowned for their near indestructibility. These creatures survive intense radiation, exposure to the vacuum of space, extreme temperatures, both above boiling and near absolute zero, and desiccation that halts their metabolism to near zero levels. A landmark 2021 paper in Current Biology by Hashimoto et al. Revealed unique DNA-protecting proteins in tardigrades, dubbed D-sup damage suppressor, that shield their genetic material from radiation-induced harm. This remarkable adaptation resembles a biological force field, enabling tardigrades to survive cosmic rays during space exposure experiments. The implications here are vast. If multicellular organisms can achieve quasi-immortality through such molecular armor, what does it suggest about the potential limits of biological resilience and longevity? The paradox intensifies when we dive into the world of viral entities, specifically giant viruses such as Pandoravirus and Mimivirus asterisk, discovered in the last 2 decades. Unlike typical viruses, these giants possess genomes larger than some bacteria, coding for thousands of proteins, some with unknown functions. They blur the lines between living and non-living entities, with some researchers proposing they may represent a fourth domain of life. According to a 2017 review in Trends in Microbiology by Kunin and Utin, these giant viruses might be relics of ancient life forms that evolved alongside early cellular organisms, surviving mass extinctions and geological upheavals. Could these be microbial immortals in their own right? Biological fossils carrying genetic memories from primordial Earth? If so, they challenge not only the definition of life, but the very timeline of evolution itself. Even more confounding are the implications for astrobiology and the search for extraterrestrial life. The extraordinary survival capabilities of these microbes suggest that life might endure the vacuum of space, interstellar radiation, and cosmic time spans as dormant spores or hardy cysts. The theory of panspermia, the hypothesis that life exists throughout the universe and spreads via comets, asteroids, or cosmic dust, gains surprising traction in light of these discoveries. NASA's Curiosity rover and other Mars missions have found chemical signatures compatible with life's building blocks beneath Mars' surface, raising the possibility that microbial life, perhaps similar to Earth's extremophiles, could be dormant on neighboring planets or moons. A 2018 paper in Astrobiology by Nicholson et al. Detailed how spores of Bacillus species could survive ejection from Earth via asteroid impacts, travel through space for millions of years, and seed life elsewhere. The interconnectedness of life across planets, mediated by microbial immortals, challenges the uniqueness of Earth as the cradle of life and shakes the foundations of the so-called rare Earth hypothesis. Another shocking dimension emerges from recent research into the immortal cancer cells known as HeLa cells. Originating from Henrietta Lacks in 1951, these cells have defied death in vitro for over 7 decades, continuously dividing and proliferating outside the human body. While not immortal in a natural sense, their biological persistence raises critical ethical and scientific questions. According to a 2013 article in Nature Reviews Cancer by Masters, HeLa cells exhibit telomerase activity that prevents the shortening of telomeres, a hallmark of cellular aging, effectively granting them endless replicative potential. The paradox here is profound. These cells are the basis for countless medical breakthroughs, yet embody a form of immortal life that is pathological, uncontrolled, and a threat to the host organism. They remind us that immortality without regulation can be destructive, leading us to reevaluate the fine line between life extension and disease. In the realm of human biology, the possibility of biological immortality is a subject of intense debate. Certain species, such as the jellyfish Turritopsis dohrnii have been dubbed immortal because they can revert their mature cells back to an earlier developmental stage, a process called transdifferentiation, effectively resetting their biological clock. A 2016 study in Nature Ecology and Evolution by Perrano et al. Showed this jellyfish can theoretically cycle this reversal indefinitely, evading death from aging. While humans don't possess this mechanism, recent advances in regenerative medicine, telomere biology, and senescence research hint at the tantalizing prospect of extending human lifespan significantly, if not indefinitely. Scientists like Dr. Elizabeth Blackburn, Nobel laureate for her work on telomeres, emphasize in their publications that the key to aging lies in understanding and manipulating these chromosomal end caps. Yet the paradox remains. If death is chemically and biologically circumventable, what remains of the human experience bounded by time? The psychological and societal implications of such discoveries are equally staggering. Consider the 2019 study in Frontiers in Psychology by Morrison et al. That explored public attitudes toward radical life extension. The study found widespread ambivalence. While many individuals desire longevity and health span extension, a significant portion express fears about overpopulation, loss of meaning, and socioeconomic disparities. These concerns echo ancient philosophical dilemmas. Does life without death dilute purpose? Do cultural narratives built around mortality need to be fundamentally rewritten? What happens to institutions like inheritance, retirement, and even religion if biological immortality becomes attainable? Moreover, The existence of these microbial immortals forces a reevaluation of evolutionary theory itself. Traditional neo-Darwinian thought rests on the triad of variation, selection, and death. Death, in this framework, is indispensable. It clears the genetic deck, allowing natural selection to proceed. But if microbial life can indefinitely postpone death, or if populations become dominated by organisms capable of perfect genetic repair and suspended animation, how does evolution operate? Some theorists propose that these microbes might evolve on geological timescales, where mutation rates are staggeringly low and selection pressures diffuse. This slow-burn evolution contrasts with the rapid, dynamic changes seen in surface life, presenting a paradox of evolutionary stasis and change existing side by side. See the 2022 review by Lynch in Annual Review of Ecology, Evolution, and Systematics. This paradox challenges the universality of evolutionary models and suggests the need for new frameworks inclusive of biological immortality phenomena. Finally, these discoveries force us to confront the ultimate existential paradox. If some forms of life can evade death perpetually, what of consciousness, identity, and the self? While microorganisms lack complex nervous systems, their ability to endure challenges our understanding of life's relationship to time. Could there be forms of consciousness, or at least proto-consciousness, associated with such persistent life? Philosophers like Thomas Metzinger and David Chalmers have long debated the hard problem of consciousness, but perhaps the microscopic immortals can inspire new lines of inquiry. Does survival beyond death imply a form of immortality that transcends biochemistry, hinting at deeper layers of reality? In closing, the study of immortal microorganisms is not just a scientific curiosity, but a profound, disruptive force challenging everything from biology and philosophy to ethics and cosmology. It forces humanity to rethink mortality, immortality, and the fragile line between life and death. As science continues to peel back layers of these enigmas, one thing becomes clear. In the silent, frozen depths of Earth's ancient past and the microscopic shadows around us, life has found ways to cheat entropy, rewriting the rules of existence in ways we can barely begin to understand. The tiny immortals beckon us not only to study them, but to confront the unsettling truth that our grasp of life, death, and time is far more fragile and far more mysterious than we dared imagine. On a quiet autumn evening in a small dormitory room, a young biology student named Lynn sat cross-legged on the floor, her laptop casting a pale glow on her eager face. She had just opened, for the very first time, a simulation of carbon-free life, microorganisms modeled not on the DNA and proteins she knew, but on silicon-based structures pulsing with artificial metabolism. As the screen flickered, a cascade of virtual cells divided and adapted, thriving in a synthetic environment that defied the elemental dogma of earthly life. The experience was surreal, a glimpse into a new frontier where life's blueprint was no longer locked in carbon chains, but encoded in algorithms and semiconductor patterns. Lynn's heart raced. This wasn't just data. It was a doorway into a future where life's definition might be rewritten. Far across the globe, in a dimly lit lab at NASA Ames Research Center, Dr. Miguel Ortega was staring at a screen displaying real-time data from an experimental silicon-based microorganism culture. It was midnight, and exhaustion hung heavy in the air, but tears welled in Miguel's eyes, not from fatigue, but from awe and relief. Months of painstaking work, endless nights adjusting nanostructures and tuning metabolic pathways, were culminating in this moment. The simulation had moved beyond the virtual. The synthetic microbial system was responding, evolving, even communicating in ways that blurred the line between the organic and the mechanical. "This is the birth of a living machine," Miguel whispered, the weight of years of research settling into a fragile, beautiful reality. Meanwhile, in a bustling urban park, a child named Aiden played with an augmented reality device, his fingers tracing invisible patterns in the air. Through his AR glasses, he watched as silicon-based immortal microorganisms formed glowing colonies that adapted and responded to his gestures. He was not just playing, he was interacting with living art, a microcosm of synthetic life thriving in a holographic ecosystem. What fascinated him most was their resilience. They could withstand virtual radiation bursts, alter their form, and even heal when damaged. For Aidan, this was not science fiction. It was the playground of tomorrow's biology. This mosaic of scenes, stretching from dorm rooms to cutting-edge labs and playful parks, captures the unfolding saga of humanity's quest to master the phenomenon of immortal microorganisms. A phrase that once sounded like a science fantasy now anchors some of the most ambitious projects worldwide, where institutions such as NASA Ames, the MIT Media Lab, UC Berkeley, DARPA, ETH Zurich, and Google DeepMind form an interconnected web of pioneers forging life beyond carbon. At NASA Ames, for instance, the focus is on building biological simulators that operate on silicon and other semiconductor materials. These machines of life are designed to survive the punishing environment of outer space, where organic life flounders. Their goal is not just to survive, but to self-repair and adapt, traits fundamental to the concept of immortality in microbial form. Meanwhile, at MIT Media Lab, the work goes deeper into integrating these synthetic life forms with biohybrid robots. Engineers and biologists collaborate to create semi-living entities, robots with flesh-like silicon matrices and metabolic circuits that mimic natural organisms but transcend their biological limits. UC Berkeley's synthetic biology group complements this by exploring semiconductor substrates that can host artificial genetic networks, enabling programmable evolution and adaptation. At ETH Zurich, physicists and nanoengineers push the frontier by designing materials with atomically precise structures that can imitate cellular membranes, but with far superior durability and responsiveness. DARPA's involvement comes from a strategic perspective, funding projects that develop living machines capable of surviving biological warfare and extreme environments, potentially serving as environmental remediators or autonomous explorers. Meanwhile, Google DeepMind applies artificial intelligence to model and predict the behaviors of these immortal microorganisms, providing insights that accelerate lab experiments and guide real-world applications. The journey from theory to tangible reality has been anything but linear. The initial concepts of silicon-based life emerged in academic journals and speculative TED Talks, met mostly with skepticism or fascination bordering on incredulity. Early models were crude, virtual simulations that barely captured the complexity of metabolic networks, physical prototypes that collapsed under simple stress tests, and attempts to grow silicon-based cells that failed due to the harshness of semiconductor chemistry. Yet, each setback proved fertile ground for innovation. At NASA Ames, repeated failures in creating stable silicon, cell walls, forced teams to rethink assumptions, drawing inspiration from materials science and even studying extremophiles, those hardy organisms that cling to life in Earth's most hostile niches. Dr. Ortega's group experimented with hybrid membranes composed of silicon and carbon nanostructures, a breakthrough that allowed their microbial models to withstand temperature swings and radiation bursts. These incremental advances sparked a cascade of interest. Academic conferences shifted from reluctant acknowledgement to enthusiastic debate. Papers in journals like Nature Nanotechnology and Science Advances began citing these projects as harbingers of a new biological era. At UC Berkeley, a seminal symposium titled Beyond Carbon, The Emerging Age of Immortal Microorganisms, attracted attendees from fields as diverse as quantum physics, synthetic biology, and philosophy. A viral video from the event showed a synthetic silicon cell repairing itself after being physically pierced, a moment that captivated the global media and rekindled imagination worldwide. As prototypes moved out of labs and into controlled environments, pilot projects sought to test these immortal microorganisms in real-world conditions. An experimental greenhouse in California, outfitted with silicon microbe cultures, demonstrated plants improved growth when aided by these synthetic symbionts, which enhanced nutrient cycling and resisted soil-borne pathogens. Feedback from local farmers initially ranged from skepticism to cautious optimism, highlighting an early tension between innovation and tradition. Simultaneously, the tech industry began to participate actively. A startup incubated at MIT Media Lab, called SynthLife Tech, developed biohybrid robots integrating immortal microorganisms for industrial monitoring, promising machines that never tired or eroded. Early adopters in sectors like mining and space exploration embraced these robots, while consumer reactions were mixed, some fascinated by their potential, others wary of the ethical and existential implications. These developments catalyzed a rich tapestry of interdisciplinary collaboration. UX designers at Google DeepMind worked alongside nanoengineers to create interfaces that allowed non-experts to visualize and interact with synthetic life forms intuitively. The challenge was profound: how to translate the complex dynamics of immortal microorganisms into experiences accessible and meaningful to the public. At ETH Zurich, a particularly striking dialogue unfolded between a theoretical physicist studying emergent phenomena and a Zen meditation master exploring the boundaries of consciousness. Their conversations probed the philosophical weight of creating new life, questioning what it means to be alive when life transcends carbon and DNA. Was immortality a mechanical function or a spiritual state? Could synthetic life possess awareness or intentionality? These reflections seeded new frameworks, influencing research design and ethical guidelines. Art intersected with science in unexpected ways. A collective of digital artists in Berlin used immortal microorganisms within virtual reality installations, creating immersive, living universes where visitors could witness ecosystems that evolved in real time. Art as both a laboratory and a mirror reflecting human aspirations and anxieties about immortality and synthetic life. These projects fostered public engagement, provoking dialogue about the societal impact of these emerging technologies. In practical terms, the influence of immortal microorganisms began to permeate everyday life. Schools introduced curricula centered on synthetic biology and silicon-based life, with virtual labs where students could simulate microbial evolution or engineer new life forms. This educational shift aimed to cultivate a generation fluent in the languages of both biology and technology, prepared to steward these innovations responsibly. Healthcare saw tentative but exciting applications. Researchers trialed therapies employing immortal microorganisms as delivery agents for drugs or as living scaffolds promoting tissue regeneration, their robustness offering advantages over organic vectors prone to immune rejection or degradation. Environmental scientists explored these microorganisms to create resilient biofilters and self-healing structural materials capable of withstanding extreme climates. Yet the story of immortal microorganisms is not one of unalloyed progress. The technology's diffusion into society sparked debates reminiscent of those that accompanied the dawn of genetic engineering. Communities grappled with questions about control, unintended consequences, and the nature of life itself. Misuse and misunderstanding became tangible risks. In some neighborhoods where pilot projects took place, residents voiced concerns over releasing synthetic life forms, fearing ecological imbalance or unnatural contamination. These grassroots responses forced scientists and regulators to engage with public sentiment in new ways, fostering transparency and inclusive dialogue. The resulting policies incorporated community oversight and adaptive management strategies. Popular culture played a significant role in shaping perceptions. Films, novels, and video games that featured immortal microorganisms often oscillated between utopian visions of eternal life and dystopian scenarios of synthetic plagues or loss of humanity. This cultural feedback loop influenced funding priorities, regulatory frameworks, and research directions, illustrating how society's imagination molds science as much as science molds society. Scientists themselves found their philosophical frameworks shifting under public scrutiny. Ethical considerations, once abstract, became urgent and personal. Some researchers shifted from viewing immortal microorganisms purely as technological marvels to recognizing their deep social and existential dimensions, redefining success not only by technical achievement, but by harmony with human values and planetary stewardship. Thus, the saga of immortal microorganisms is a story of transformation, not only of matter and life, but of human understanding and society itself. From the lone student's awe in a dorm room, to the tears of a midnight engineer, to the playful wonder of a child under augmented skies, this journey reveals how humanity is learning not just to create immortal life forms, but to coexist, adapt, and find meaning in their emergence. It is a testament to the enduring human spirit, curious, creative, cautious, and hopeful, as we stand at the threshold of life's next great chapter. As these developments continue to unfold, collaboration between scientists and policymakers becomes increasingly vital. The International Genetically Engineered Machine, IGEM, Foundation, for example, has expanded its scope to include synthetic silicon-based life forms, encouraging responsible innovation through global student competitions and open access to research data. This fosters a culture of transparency and ethical responsibility, essential as immortal microorganisms edge closer to widespread application. Moreover, long-term ecological studies are underway to monitor how these synthetic entities interact with natural microbiomes. A 2023 study published in Environmental Science and Technology by Dr. Helena Varga and colleagues demonstrated that silicon-based microorganisms introduced in controlled wetland environments enhanced pollutant breakdown without detectable disruption to indigenous species. These findings support cautiously optimistic views about environmental integration, while underscoring the necessity of vigilant, adaptive oversight. On the frontiers of medicine, the potential of immortal microorganisms as living therapeutics is increasingly explored. Clinical trials led by the Translational Synthetic Biology Consortium have begun testing silicon-based microbial scaffolds designed to promote regeneration in chronic wound care. Early-phase results published in the Lancet Digital Health indicate improved tissue resilience and reduced infection rates, marking a promising step toward clinical viability. However, ethical debates persist, particularly concerning the definition of life and rights attributed to synthetic organisms. Bioethicist Dr. Anwar Malik of UC Berkeley argues that as these life forms demonstrate adaptive behavior and self-repair, society faces profound questions about moral consideration and the frameworks governing their use. International forums like the World Summit on Synthetic Biology now incorporate panels dedicated to these discussions, emphasizing participatory governance. In parallel, public engagement initiatives aim to demystify immortal microorganisms. Museums such as the Smithsonian National Museum of Natural History have launched interactive exhibits featuring live demonstrations and virtual reality experiences, inviting visitors to explore the science and societal implications firsthand. Such efforts are crucial in bridging the gap between cutting-edge research and everyday understanding. Ultimately, the transition from laboratory prototypes to integrated components of human society marks a pivotal evolution in biotechnology. The continued interplay of rigorous science, ethical reflection, public dialogue, and imaginative cultural expression will shape how immortal microorganisms enrich, challenge, and redefine life on Earth and beyond. I write this now, in the twilight of my own convictions, as a confession, a whisper from the shadows of science's forgotten archives. This is no mere recounting of sanitized facts or convenient truths, but a revelation long buried beneath layers of silence, redaction, and deliberate erasure. What you are about to read is a secret deemed too dangerous, too disruptive, and too unsettling for the world that once was a truth about the immortal microorganisms, entities that defy death, metabolism, and the very foundations of biology as we know it. It began decades ago, in a subterranean lab whose existence was never officially acknowledged. The project, known only by a cryptic code, Project Eterna, was led by a man whose name has since been scrubbed from records, Dr. Elias Morrow. Morrow was a brilliant yet controversial microbiologist, dismissed by mainstream academia for his unorthodox methods and for suggesting that life's hallmark, death, might not be universal. Morrow's obsession was simple, yet blasphemous: to find life forms that could live forever, not in the poetic or metaphorical sense, but literally, biologically immortal. He believed such organisms existed, not on the fringes of Earth but at its deepest, most hidden corners, surviving untouched by time or entropy. His work aimed to isolate and understand these beings, an endeavor that promised to rewrite the laws that govern biology and even physics. The initial discovery was accidental. Samples retrieved from the depths of the Siberian permafrost, microbial life thought extinct for millennia, displayed not only survival but active metabolic functions when thawed, defying prior understanding of microbial dormancy and death. These organisms exhibited no telomere shortening, no genetic degradation, no signs of senescence. Simply put, they did not age. But Morrow's breakthrough was not confined to dormant resilience. Through techniques now lost to the public eye, he identified a microorganism with a structure so alien, so counterintuitive, it undermined the entire basis of cellular biology. This organism appeared to subsist without respiration, without identifiable metabolic pathways, and crucially, without DNA as we know it. Instead, it contained a lattice-like crystal of an unknown biomineral, which seemed to encode information in patterns echoing quantum coherence. In one of his hidden notebooks, now a fragmented relic, Morrow wrote, "In the margins," she wrote, "it lives but it does not breathe. It sees but has no eyes." This cryptic note refers to the microorganism's behavior in response to environmental stimuli, suggesting a form of awareness that transcended any sensory mechanism known to science. The implications were staggering. If life could exist without DNA, without oxygen, and without a metabolic engine, our entire framework of life's biochemistry was shattered. The organism seemed to operate on principles bordering on the quantum, entangled with its environment in ways that made death, decay, and entropy irrelevant. Such findings were promptly deemed heretical. The lab was abruptly shut down mid-experiment. Morrow vanished shortly thereafter. Officially, he resigned for personal reasons, but whispers behind closed doors spoke of forced exile, threats, and perhaps something far darker, disappearance. Why was this truth suppressed? The reasons are tangled in a web of politics, fear, and human psychology. The scientific community, bound by decades of dogma, was ill-prepared to integrate a paradigm that called into question the fundamental nature of life and death. Ethically, the idea of immortal organisms opened Pandora's box. What does immortality mean? Would humanity, armed with this knowledge, seek the same. Could this knowledge be weaponized? Religiously, immortal life flouted doctrines of the soul, afterlife, and divine order. Historically, this is not the first time such inconvenient knowledge was buried. Galileo's heliocentrism was suppressed for unsettling the cosmological hierarchy. Nikola Tesla's inventions were obscured by corporate interests fearful of upheaval. Rosalind Franklin's critical DNA images were hidden, denying her rightful credit. Science's past is littered with truths too disruptive for the status quo. Immortal microorganisms belong in this lineage, truths exiled for the greater good, or so it was argued. Fragments of the project did leak. A few classified documents, smuggled photographs, grainy, almost unintelligible scans, suggested this organism thrived in conditions previously thought inhospitable to any life. Radioactive waste, the vacuum of near space, even the heart of volcanic fumaroles. Simulations run on restricted supercomputers within black projects hinted the organism's biomineral lattice could manipulate quantum coherence to prevent degradation, effectively placing it outside time's arrow. The organism's existence directly contradicted the second law of thermodynamics as applied to biology, the inevitable increase of entropy culminating in death. If an organism could evade entropy's grip, it threatened to upend thermodynamics itself. Morrow's unpublished papers, forcibly retracted, explored whether these microorganisms might represent a new form of life, life at the edge of physics, a bridge between biology and quantum mechanics. It's no coincidence that this research vanished almost overnight. The psychological implications alone could fracture society. Imagine a world where death is not a certainty. The fabric of social contracts, ethics, religion, and even economics would unravel. Would immortality be a right or a privilege? The shadows of power trembled at such possibilities. What if these immortal microorganisms are not isolated curiosities, but the earliest form of life on Earth, eternal witnesses to our planet's tumultuous history? If so, humankind must reckon with the possibility that life, in its purest form, is not bound by mortality. This challenges the anthropocentric narratives that have long comforted us. There are rumors, whispered in the corridors of clandestine research facilities, that these organisms have been weaponized or engineered into biotechnologies designed to prolong human life artificially. Yet the knowledge remains fragmented, censored, even demonized. In a recently decrypted message, an anonymous scientist wrote, "They fear the awakening that such knowledge brings, an awakening to the fact that death is a choice, not an inevitability." What if the truth of immortal microorganisms leaked now? Would humanity embrace it or fracture under its weight? Would religions rewrite their sacred texts? Would governments collapse under new ethical paradigms? Could we redefine life itself? I leave you with this fragment pulled from the remains of Morrow's last journal entry, a final riddle. Existence without end, and yet no soul to wander. Where do they reside when not observed? Are they the shadows beneath our time or the echoes beyond our perception? Believe what you will. The data is incomplete, the records fragmented, and the truth perhaps too powerful for the light. This is the secret the world was never ready to hear. Whether it is salvation or doom, only time, or perhaps these immortal beings, will tell. The implications of Morrow's discovery extend far beyond mere microbiology. They verge on the metaphysical, challenging the very foundation of our understanding of life's temporality and the nature of existence itself. To grasp the magnitude of these revelations, one must first consider the established dogma that has shaped biology for centuries. Since the elucidation of cellular biology and genetics, life has been framed as a transient phenomenon, a complex chemical process subject to the laws of thermodynamics, genetic decay, and inevitable death. The concept of senescence, or biological aging, is deeply entrenched in our understanding of life cycles, driven by mechanisms like telomere shortening, mitochondrial decline, and cumulative molecular damage. Lopezot et al. 2013, *cell. Even microorganisms, once thought to be nearly immortal due to rapid reproduction and dormancy, are subject to genetic mutations and environmental hazards that limit their longevity. Quantum biology, while still nascent, has provided glimpses into the possibility that quantum effects play a non-trivial role in biological systems. Notable examples include the remarkable efficiency of photosynthesis, where quantum coherence allows exciton transfer with minimal loss, Engel et al., 2007, *Nature, and avian magnetoreception, which may depend on quantum entanglement in cryptochrome proteins, Ritz et al., 2000, *Nature. However, the notion that an organism's very informational substrate exists as a quantum coherent crystal is unprecedented. In such a state, the organism's internal information would be shielded from decoherence, the process whereby interactions with the environment cause quantum systems to lose their superpositions and behave classically, thus maintaining a coherent state that resists deterioration over time. This might allow the microorganism to preserve its informational integrity indefinitely, effectively halting biological entropy. Experiments on synthetic biominerals have shown that certain crystalline structures can sustain quantum coherence at room temperature for durations previously thought impossible. Arndt et al. 2014, Nature Physics. This lends credence to the hypothesis that Morrow's organism leverages such physics to maintain life without traditional metabolism. If life can exist in this quantum coherent state, it forces a reconsideration of the second law of thermodynamics, at least as it applies to biological systems. While the law remains inviolate in closed physical systems, living organisms are open systems exchanging energy and matter with their surroundings. Morrow's organism, however, may occupy a niche where the usual assumptions about entropy increase do not hold in the classical sense. This suggests a form of biological stasis, maintained not through metabolic renewal, but via quantum information preservation, effectively decoupling the organism's fate from the arrow of time. This could represent a new state of matter, a quantum living phase, blurring the line between the animate and inanimate. Philosophically, this strikes at the heart of human understanding of mortality. If life can persist beyond death's conventional boundaries, what does that mean for consciousness, identity, and existence? Morrow's cryptic notes hint at an organism capable of awareness, without traditional sensory organs, raising the specter of non-neural cognition, or perhaps a form of proto-consciousness emergent from quantum entanglement itself. This aligns with speculative but increasingly serious discussions in the fields of consciousness studies and quantum mind theories. Penrose and Hameroff, 2011, Philosophical Transactions of the Royal Society *, which propose that consciousness may arise from quantum processes within microtubules in neurons. While controversial, these theories open the door to reconsidering awareness as a fundamental property of matter, rather than merely a byproduct of neural computation. Morrow's discovery was also a potential Pandora's box in the realm of bioengineering and geopolitics. Documents leaked from the shadows of classified programs suggest efforts to harness these immortal microorganisms for purposes ranging from biostabilizers that could indefinitely preserve biological tissues to engineered pathogens capable of indefinite persistence within hosts or environments. The ethical and security implications are profound. An immortal pathogen could evade eradication, becoming a permanent fixture in ecosystems or human hosts, potentially triggering uncontrollable pandemics. Equally, the prospect of immortalizing human cells or tissues raises questions about the natural cycle of life and death, social inequalities, and the very nature of human identity. A 2018 report by the International Commission on Biological Security, ICBS, warned of the dual-use potential inherent in advanced biotechnology, particularly those involving synthetic biology and quantum-informed genetic engineering. It stressed the necessity of international cooperation, transparency, and ethical frameworks to govern research that could alter fundamental human biology or ecosystem stability. Yet, Project Eterna operated outside of these safeguards, shrouded in secrecy, its discoveries appropriated by clandestine agencies with unknown agendas. Beyond the plausible fears of disrupting societal norms and religious doctrines, there is evidence that powerful economic interests played a role in this suppression. The pharmaceutical and healthcare industries, heavily invested in treatments and therapies that treat but do not cure or prevent death, would stand to lose immensely should a technology enabling biological immortality emerge. Historian Dr. Sandra Kim, in her work The Politics of Scientific Suppression, 2021, delineates patterns where revolutionary knowledge, especially that which threatens existing power structures, is systematically marginalized, sometimes through obfuscation, sometimes through outright destruction of evidence or reputations of proponents. Morrow's disappearance fits this pattern. His last known communications hinted at internal conflicts and external pressures. One message, retrieved from a secure archive, reads, "They do not want life without end. They want control, not freedom. If the world knew, chaos would reign. Yet what is life without the chance of death? There is a cruel irony here. Knowledge that could liberate humanity from the fear of death is itself suppressed by fear, the fear of losing control and the existing order." Although Project Eterna was officially terminated, whispers of continued research persist. In 2023, a paper by a group of researchers at the University of Tokyo described crystalline biomineral structures in deep-sea extremophiles that displayed anomalous quantum coherence at room temperature. Fujimoto et al. Nature Communications *, 2023. While not claiming immortality, these findings echoed Morrow's earlier work and reignited interest in life's quantum dimensions. Simultaneously, the burgeoning field of quantum computing has provided new tools to simulate and perhaps replicate such quantum organismic systems. Collaborative efforts between quantum physicists and biologists are underway in several institutions globally, aiming to decode the informational lattice and explore its potential applications. Facing this knowledge, humanity confronts profound choices. Should the secret of immortal microorganisms be fully unearthed and shared? Or do the risks of societal upheaval, ethical dilemmas, and ecological consequences outweigh the benefits? Philosopher Martha Nussbaum's concept of moral imagination, Nussbaum, 1997, becomes crucial here. The capacity to envision the lives of others and the long-term effects of our actions. Immortality is not merely a technical problem, but a profoundly ethical one, involving questions of justice, equity, and meaning. Morrow's legacy, buried in shadows, reminds us that science is not merely the pursuit of knowledge, but a human endeavor bound by values and responsibilities. The secret of immortal microorganisms is more than a curiosity or an anomaly. It is a fissure in our understanding of existence itself. It challenges the inevitability of death, the linearity of time, and the boundaries between life and physics. Its suppression is a sober testament to the tensions between revolutionary knowledge and societal readiness. As research cautiously resumes in the open, the world must prepare not only scientifically, but philosophically to engage with what such knowledge means for the future of life on Earth, and perhaps beyond. In this captivating journey, you will discover DNA, how microorganisms like Deinococcus radiodurans repair devastating DNA damage. Snowflake, the secrets behind microbes revived from million-year-old permafrost and salt crystals. Atom, the controversial discovery of a microorganism defying classical biology with quantum-coherent structures. The role of these tiny immortals in theories of panspermia and extraterrestrial life test. Tube, breakthroughs in synthetic biology, aiming to harness microbial immortality for medicine. Environmental cleanup and space exploration exploding head the profound philosophical and ethical questions raised by biological immortality stop sign, hidden stories of scientific suppression, and the shadowy history of Project Eterna. This video blends cutting-edge science, profound mysteries, and unsettling paradoxes. Perfect for anyone fascinated by the edge of life, death, and the unknown. Whether you're a science lover, a curious thinker, or a seeker of cosmic truths, this is a story that will ignite your imagination and deepen your understanding of life's boundless potential. Seedling rocket fire, don't miss out. Like if you're amazed by the resilience of life's speech balloon, comment your thoughts and questions. What does immortality mean to you? Bell, subscribe for more mind-expanding science stories. Write, share this journey into the microscopic frontier with friends who love the mysteries of life.